Uh, so we are live on both Facebook and YouTube, and we are joined by a lot of people now. And I'm just accepting people in the meeting. So we will have to start. It is 7 p.m. Bangladesh Standard Time, and 2 p.m. British Summer Time, 1 p.m. I guess in UTC. So it's 6:30 p.m. in India Standard Time. So we are going to start. Dr. Abdullah Mawashwal, sir, can we wait for like 3-4 minutes? I guess there will be a lot of people joining us.
I think better you should continue, and you should introduce Science Portal BD and activities.
Okay, sir. Okay,
sir. People will join. After that, you will start, please.
Oh, okay, sir. We are getting a lot of requests. Okay, I'm accepting 11. Okay. Mage. Yep. Okay, so let's start. Bismillahirrahmanirrahim. In the name of Allah, the most gracious, the most merciful. We'd like to start shipping the future virtual seminar on CRISPR-Cas 9, entitled CRISPR-Cas 9, New Frontiers Technology for Future Agriculture. Assalamu alaikum, adab. Good morning, good afternoon, good evening, everyone, depending on wherever you are watching us live on Google Meet, Facebook, and YouTube. I'm your host, Sam Shurkar from Bangladesh. Welcome to the seminar. So there are 3 primary ways you can participate in today's event. The first, 80 registered people have joined us to the panel by Google Meet. Others are watching us live on Facebook and YouTube. So if you face any difficulties in one of the platforms, please consider trying another. For Facebook, please visit tiny.cc/SPB12, SPB13 for Facebook, or you can watch us live from SciencePortal Bangladesh's official Facebook page at fb.com/SciencePortalBD. For YouTube, please visit tiny.cc/SPB12. You can find all the links at SciencePortal Bangladesh's official Facebook page at fb.com/SciencePortalBD. Dear Google Meet attendees, please make sure to keep your video and microphone turned off other than the panel. And for panel, please make sure to mute your microphones other than the speaker so we do not get any background noises and participants have no difficulty understanding us. Dear attendees, please put on your headphones for the best ever experience. But still, if you face any difficulties whatsoever, please let us know in the comment sections. We'll try to fix them as soon as possible. First of all, I'd like to thank our partners and sponsors. Shipping the Future Virtual Seminar on CRISPR-Cas 9 is brought to you by SciencePortal Bangladesh, sponsored by Cell Genetics and Plant Biotechnology Lab. SciencePortal Bangladesh is a scientific communication platform based in Bangladesh. Though we started locally, we have been successful to deliver the message of science to more than 150,000 people from more than 65 countries. We are hosting international seminars regularly, just like this one, with some of the best scientists in the world on cutting-edge technologies ranging from precise gene editing, algal biofuel production, speed breeding, vaccine development, plant metabolic pathway hacking, GM crops, to spirulina, the feed of the future, that are free and open for all. Our members are highly enthusiastic to explore and do science. We believe anyone can do science with the right skill set and knowledge. We do not know where the next biggest breakthrough is going to come from. That's why we are promoting frugal science, a brilliant philosophy that inspires the development and distribution of cost-effective, affordable scientific tools such as Foldscope, a powerful microscope that costs only 50 cents, but is as powerful as a laboratory microscope that anyone can use regardless of their educational backgrounds. We are trying to take science out from the laboratory to the streets, to the people, where it belongs. We strongly believe that science is for everyone in its true sense. That is why we are engaging with the people at the grassroots level to make them aware of the modern innovations, to meet the global challenge of climate change, renewable energy, green planet, and sustainable agriculture, public health issues. We're educating people to achieve net zero carbon emission by the second half of the century, a goal set in the Paris Agreement in 2015. As you can guess, it is not possible for us to do all of these alone. We need your help. If you want to spread science in your community and be a part of SciencePortal Bangladesh, you are more than welcome to be a member, local ambassador, or perhaps a campus ambassador of this global family of change makers. If you're an individual with ideas or spreading, or perhaps want to share your research with a global audience, feel free to mail us at speakers@sciencebotalbd.com. If we're an organization striving for the same goals as we are, please feel free to reach out to us at info@sciencebotalbd.com. We're open to collaboration with other interested groups and people who are striving for a common goal from all around the world. This has been our 9th international initiative. Our very first seminar was on genome editing, entitled The Basics of Genome Editing What, Why, and How by Dr. Kijibin Alimullah. In fact, Dr. Mullah is with us today. We are going to have a great time together. Thank you, Dr. Mullah, for being here today. And our second seminar was on the production of fourth-generation biofuels, entitled Microalgal Cell Factories, the fourth-generation perspective for industrial biofuels by Dr. Kamoun, followed by another successful one on speed breeding technology entitled Speed Breeding Crops to Feed 10 Billion by Dr. Lee Hickey, followed by another successful one on vaccine development entitled Virus-like Particle Vaccines Made in East Against Polio and Emerging Diseases, which is covered by Professor Dr. Nicola Stonehouse, fellow of both the Royal Society of Biology Nurse and the Transit Expression for Vaccine Production Plans by BBSR's Innovator of the Year 2012, Professor Dr. George Lomonosov. Our fifth seminar was on hacking plants entitled Hacking of Plant Metabolic Pathway for the Biosynthesis of Human Therapeutic Proteins by Dr. Shashi Kumar, followed by another successful one on GM crops entitled The Next Generation of GM Crops to Address Public Health by Chief Executive Scientist Dr. Russell Rinke and Dr. Ines Lamet, followed by another successful one on the Feed of the Future entitled Spirulina, the Feed of the Future by Dr. Bijoy Chade and Mr. Rohit Shankar Mane. Our very last seminar was on the development of COVID-19 vaccines by Dr. Kathleen Heffron, scientist at Cornell University, and Dr. Kandakar Mehdi Akram, a research associate at the University of Sheffield. All of them were highly praised by the scientific community as well. You can watch the recorded video of the first 4 events on our Facebook page and our YouTube channel. The last 4 are in post-production, will be available on YouTube and Facebook within a couple of weeks. So stay tuned for that. If you want us to host more seminars like this one, then please make sure to like and follow our official Facebook page at fb.com/SciencePorterBD. Subscribe to our YouTube channel at tiny.cc/SPB_YouTube. And hit the notification bell icon to be the first to get the latest updates on our upcoming seminars. We are lucky to have Cell Genetics and Plant Biotechnology Lab as our sponsor for this event. If I talk a little bit about them, they are doing an incredible job in terms of research and are open to collaboration with other interested groups and entities from all around the world. I'd like to thank all the organizations that have cooperated fully throughout our promotion. I'd like to thank our university partners, content writers, translators, social media teams, streamers, and print media partners. And finally, I'd like to thank you, the audience. We were overwhelmed by responses. We have received around 285 registrations from all around the world. Without your continued support, we couldn't be here today. It would not be possible without the active contribution of Science Party Bangladesh team. So a big round of applause for you guys as well. Without further ado, let's introduce you to our panel. Let's meet the event chair, Dr. Abdullah Mohammad Shoail, Professor of Biotechnology and Genetic Engineering at Jahangirnagar University, Bangladesh. Dr. Shoail is a respected authority in the field of biotechnology, but for those of you who don't know him, he is the director of Cell Genetics and Plant Biotechnology Lab that is well known for its higher research output. He did his PhD in agricultural biotechnology from Chungbuk National University in South Korea. He had worked as a research collaborator for the International Rice Research Institute in the Philippines. He was awarded the prestigious JASPS postdoctoral fellowship. He had worked as a postdoctoral research associate at the Cell Genetics and Citrus Incredible Lab at the University of Florida. Dr. Shoail has published articles in many reputed international journals. And now, the attraction for tonight's event, we are delighted to have 2 of the world's leading experts, Sophien Kamoun, senior scientist at the Science Barrel Laboratory in the UK, and Professor Tofazzal Islam, professor and director of the Institute of Biotechnology and Genetic Engineering at Bangabandhu Sheikh Mujibur Rahman Agricultural University. Welcome, both of you. If I talk a little bit about them, Sophien Kamoun grew up in Tunisia, where he developed a passion and curiosity about nature. He studied genetics in Paris and Davis, California, before working in Wageningen, Ohio, and Norwich, where he is currently a senior scientist at the Science Barrel Laboratory and a professor of biology at the University of East Anglia. He is known for his seminal contributions to our understanding of plant diseases and plant immunity, having pioneered genomics and molecular biology methods to reveal fundamental insights into the biology and evolution of eukaryotic plant pathogens. His innovative work in plant pathology has resulted in new approaches to mitigate some of the world's most debilitating and serious diseases, crop diseases. Professor Kamoun has received many awards and recognitions, notably the Quid Prize and the Linnean Medal. He was elected to a fellow. He was elected as a fellow of the Royal Society of England in 2018. And Dr. Tofazzal Islam is a professor and founding director of the Institute of Biotechnology and Genetic Engineering at Bangabandhu Sheikh Mujibur Rahman Agricultural University in Bangladesh. He obtained his BSc in Agriculture and MSc in Agricultural Chemistry from Bangladesh Agricultural University, securing the first position in first class in both the cases in order of merit. He has made outstanding contribution to expand our understanding of how a phytopathogen locates its host and proceeds infection mediated by the host-specific signals, especially by the signaling compounds. He discovered the activity and mode of actions of more than 60 novel secondary metabolites that inhibit the motility of zoospores with the potential for controlling omycete phytopathogens. So, enough intro, let's dig in. Let me give you a rough overview of today's seminar. First, our speakers will talk about the potential use of CRISPR-Cas in agriculture. They will talk about the latest developments made in CRISPR-Cas technology, their scopes and limitation in great detail. They are also going to highlight the genetics of key traits like drought tolerance and disease resistance and how to exploit them using CRISPR-Cas9 to develop crops with improved traits. In the end, they'll address some of the ethical concerns and safety issues related to the use of this technology. Then our esteemed panelist, Dr. Kitibidin Alimilla, scientist at NRRI, that is National Rice Research Institute of India in Kutak, and Dr. Birhanuddin, Associate Professor of Biochemistry and Molecular Biology at Jahangirnagar University, will give their remark on tonight's presentation. And then our event chair, Dr. Abdullah Mohammed Shawal, sir, will get you with his message. In the meantime, our team will collect your questions and we will go for a Q&A session shortly. So make sure to leave your questions in the comment sections of Google Meet, Facebook, and YouTube Live ahead of time. We are really grateful to all of you for being here today. It means a lot. Thanks to all of you once again. That's all from my end. Now I will request Dr. Tofazzal, sir, founder director of the Institute of Biotechnology and Genetic Engineering at Bangabandhu Sheikh Mujibur Rahman Agricultural University in Bangladesh to power up his presentation. Dr. Tofazzal, sir.
Excellent. So I am really outstanding introduction. I am really very much excited today to meet with Sophien Kamoun, one of the best friend and collaborators, and I feel that he is one of the outstanding biologists and who have been contributing significantly in molecular plant microbe interactions. We have been known each other since long through UMICT Genomics and Genetics Network, and I am very much thankful, and I should mention here that he significantly contributed to our country through wheat blast research. He practically first, knocked me to work on the genetic identity, determination of genetic identity and origin of the wheat blast when it first emerged as a devastating disease in 2016. So Sophien, congratulations. He amazingly supported us and visited Bangladesh for a week with another big scientist, Professor Nicholas Talbot. So Sophien's presence is really different today because Sophien has lots of fans, young fans in Bangladesh. Welcome to Sophien again, virtually to Bangladesh. And I am very much thankful to Syam Sarkar for nicely introducing us, and especially the Science Portal Bangladesh, which have been doing fantastic job, incredibly a powerful job in scholarly communications, enhancing the biotechnology and frontier science through some world-class meeting, inviting through the several great leaders across the world. And I'm very much thankful to Professor Abdullah Mohammed Shuhail. He is very close to me. I love him very much. He is a JSPS fellow. We worked together for a long time. A brilliant professor at University of Jahangirnagar University. I feel so honored to be here as a speaker. And I am very much thankful and very excited to see Dr. Kutubuddin Mullah, who is one of my collaborators. We are working together to edit a book, CRISPR-Cas methods. Dr. Burhan and many others welcome to this webinar and virtual meeting. And good evening, good afternoon, good day, good morning, because people are participating from across the world, and it is truly a democratic world, amid COVID-19. I would like to show some of the slides on the CRISPR-Cas CRISPR-Cas technology and its potential for making a new revolution in agriculture. Actually, I am a fan of this technology, and Sophien is the man who actually, in fact, induced me to engage CRISPR-Cas technology. I shall tell you more detail later on. So I am a fan of this technology. I am a learner of this technology. I try to give a broad overview how this technology could be utilized in Bangladesh. And it is really a game-changing technology, one of the sharpest technologies so far discovered in the biology. It received the Nobel Prize in Chemistry. So I welcome all of you to my presentation. I am going to show you some of the slides that I prepared, and then we can discuss more. And obviously, if I miss anything, Sophien is there, and he will cover everything, because Sophien is the first man who applied CRISPR-Cas technology in plant. I saw 3 papers published simultaneously in Nature Biotechnology, and Sophien was one of the team leaders. Can you see the slide?
Yes, sir. We can see your slides.
Okay, excellent. So my talk is CRISPR-Cas technology, a potential toolkit for future food security. CRISPR-Cas is really a craze, one of the most talked issue in the biology. Bioediting, I can say it a toolkit, not only a technology, and food security is really a big challenging issue. Before start my talk, I would like to congratulate the winners of Nobel Prize in Chemistry, Professor Emmanuel Charpentier and Jennifer Doudna. They developed this revolutionary, game-changing genome editing technology, CRISPR-Cas technology. And this year, another Nobel Prize, Peace Prize 2020, received by the World Food Program. And food is really very, very important. And they said, until the day we have a medical vaccine, food is the best vaccine against the chaos. Chaos means this COVID-19 pandemic. And food obviously is a medicine for every day, because without food we cannot survive. So I would like to share the quote of Alfred Nobel. He said, "I would rather take care of the stomachs of living than the glory of the departed in the form of monument." So without food we cannot survive, and food, to ensure the security of food for the population in this earth has been a big problem and challenge in every ages of the human civilization. Agriculture has around 10 to 12,000 years. In that long journey, always we had a big challenge to ensure the food security. And you would be wondered to know that now on this earth we have 7.3 or 7.2 billion people. Nearly 2 billion people are going to bed every night with hungry stomach. This is really very bad, and with hungry stomach we cannot consider and we cannot expect and dream a peaceful world. And I would like to salute another big guy, the father of the Green Revolution, Norman Borlaug, because in mid-19th century, he made a big revolution in agriculture, which is called Green Revolution. What is a green revolution? Obviously, all of the biologists, it very well. Green revolution refers to a great increase in crop production in the second half of the 20th century through the use of fertilizers, the use of agrochemicals, cultivation of high-yielding crop varieties, and application of that means mechanization in the agriculture. And this is a very simple graph I always show, and I prepared it from the population data available in the Wikipedia. If you look at this graph, you can see the world population was almost static until 16th or 17th century, and it was due to the natural control system, famine, different diseases, and other epidemics, and human was vulnerable to that. And at that time, great economist Malthus, he proposed a theory that human population increases geometrically and food production increases arithmetically. This is why famine and other causes will naturally control the human population. But two revolutions in the human history I should mention here. One was Industrial Revolution, another one a Green Revolution. Green Industrial Revolution started in Europe, and Green Revolution started from Mexico. These two revolutions, empowered human being significantly, and population, you can see after these two revolutions, population is increasing very fast on this earth, and we have now 7.3 billion people on this earth. But these two revolutions also brought some new challenges. One is the climate change, one of the most talked issue worldwide due to global warming. And Green Revolution, I mentioned the use of lots of inputs, synthetic inputs like agrochemicals and others created serious problem in the environment. So soil health deteriorated, our environment pollution is now a big threat for sustainable crop production in this earth. And Green Revolution also considered as one of the threat of the loss of biodiversity. So we have to continue our research to address these big challenges, that is climate change, as well as the consequences of Green Revolution. One of the important points is that Green Revolution helped us to dramatically increase the food production. But the technology used in Green Revolution for increasing the food production, they are not responding now. Most of the crop food production is nearly at the plateau. So we need something new technology to address this challenge. So why we need a new Green Revolution in agriculture? To ensure the food and nutritional security, we need a new revolution in agriculture, because it is estimated that by the year 2050, world population would be nearly 10 billion. And the cultivable land, arable land is decreasing very fast, and it is about 0.2 hectares per person. Now the question is it possible to ensure the future food security by using 0.2 hectare land per person to feed the 10 billion people? It is really a formidable challenge. So it is really a big challenge, and this is why we have to work on it and apply the frontier technology to make a new Green Revolution. Another important point is that the United Nations organizations has set 17 goals to achieve by the year 2030, which is known as Sustainable Development Goals. Among 17 goals, 4 goals are directly related to agriculture. The first one, end of poverty for all. A large proportion of the people are working and their livelihood are associated with the agriculture, and most of the farmers worldwide, a large proportion are associated with the poverty. So agriculture is linked to it, and freedom from hunger, as I mentioned. 2 billion people every day, they are going to bed at night with hungry stomach. So this is the number 2 challenge we have to achieve by the 2030. And other challenges like sustainable ecosystem, sustainable consumption and production, they are also linked to the agriculture. This is why we need to make a new Green Revolution in agriculture to achieve the even Sustainable Development Goals. Food security, this is a very big issue, and food is called medicine, as I mentioned. There are, what is food security? Food security has 4 pillars, like availability. Food, is the supply of food adequate? Access, can people obtain the food they need? Utilization, do people have enough intake of nutrients? And stability, can people access food at all times? So these are the pillars associated with the food security, and to ensure the food security, we have to ensure these 4 pillars. This is really a formidable and big challenge. Why food security is a challenging task? I should summarize here. To meet growing demand by 2050, a global increase in crop production, 70 to 100% is required. That means from now to 2050, we have to increase our crop production nearly 100%. But our arable land is shrinking, and it is really a big challenge. Another big challenge is climate change, which poses a serious threat to the crop production through various biotic and abiotic stresses, like incidence and emergence of serious diseases like wheat blast, or a drought, salinity, heat stress, cold stress, lots of problems that are big threat for increasing the food production. The rate of increase of yields brought about by the last green revolution has been steadily declining. This is really another alarming issue. The conventional strategies used for crop breeding are laborious, time-consuming, and complicated. For example, to develop a disease-resistant crop variety, we need to work 10 to 15 years. But this is a too slow process, and we need a faster approach. So more effective and time-saving breeding methods are required. Food security status, if I share with you in Bangladesh, because many of the participants in this webinar from Bangladesh. Bangladesh has tremendous success in food security, achieving the food security. We are nearly self-sufficient in our cereal production, but demand for food is continually increasing due to population growth. Our population growth is now 1.8% per year. And the country needs to produce more food to feed more than 2 million people that are added to the population every year, and would reach to 230 to 250 million in 2050. So these huge number of population, if we want to feed them, ensure the food security, we have to work very hard, because our land is decreasing. The most vulnerable, Bangladesh is considered most vulnerable nation on the earth to this problem, and ranked 6th on the global climate risk index 2017. That means Bangladesh is most vulnerable to climate change, and our land is very limited, and the quality of soil is heavily deteriorated due to the mining of minerals by cultivating 3 crops per year from a land. So there are some recognized major challenges associated with profitable and sustainable crop production, because our government targeted to make agriculture profitable and make agriculture sustainable for feeding the growing population of the country. But emergence of diseases and insect pests are one of the biggest challenges. For example, wheat blast, which appeared or emerged in Bangladesh in 2016, and it remarkably reduced the wheat production in the country, and Bangladesh is now 5th highest wheat importing country. Rice blast every year devastate huge amount of rice crop in our country, and our agriculture is has heavy reliance on chemical inputs such as fertilizer, water, and pesticides. This is why input cost is very high, and agriculture is no more profitable, especially in rice and other crop production. And it is a big challenge. This is why government set to make agriculture profitable. Sustainable. And extreme climate change enhances soil salinity, alkalinity, drought, heat, cold stress, and high incidence of pest and deficiency of soil micronutrients, poor elasticity in increasing desired yield, and explosion of human population, degradation of environment, and increasing scarcity of cultivable land. These are the recognized, nationally recognized challenges already specified by the government. So we have to address all these challenges by using smart or technology, and to make our agriculture as a smart agriculture. How can we make it? We need to use technology aided agriculture that minimizes the impact of climate change, breeding for adaptation to biotic and abiotic stresses. Fortification means to make our food highly nutritious, and we obviously increase the yield. And another area is domestication of wild relatives of crops or orphan crops, which is very, very interesting area, because due to the new technology, CRISPR-Cas technology, we can domesticate the relative crops, because in the all over the world, 15 to 20 species of the food crops have been used for getting the nutrition, but more than 5,000 food crops are neglected or underutilized. And if we can domesticate them and we can make them useful, then we can address the climate change consequences significantly. So one of the revolutionary technology is considered as the CRISPR-Cas genome editing, and it has huge potential to make a new green revolution in agriculture. Now, question is, what is CRISPR? You know that Dr. Kutubuddin Mullah already delivered a lecture on CRISPR-Cas organized by Science Portal Bangladesh. But I would like to just repeat some of the issue, because some new participants may be here. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. What is it? It is a sharpest genome editing technology that targets a specific section of DNA, makes a precise cut or break at the target site. Cut can do one of at least two things: makes a gene non-functional if it is a bad gene, or if it is associated with a negative trait or negative regulation. Then if we make it non-functional, then you can improve the quality of a crop. Replace one version of gene with another, and Cas is the CRISPR associated protein. So what is gene editing? Obviously, you know that gene editing is the precise alteration of genetic sequences in the living cells, including those of human, at much higher accuracy, efficiency, and ever before possible. This definition given by David Baltimore, who got Nobel Prize in 1975. We can say very simply, any techniques using site-directed mutagenesis to make small changes to DNA sequences at a known location is called gene editing and genome editing. And this game-changing method, that is CRISPR-Cas method, is discovered through a series of basic research, because in 1987 a Japanese scientist, Ishino, he found in a bacterial sequence that some fragment of DNA sequence repeatedly present in the genome of the bacteria, and it was a strange phenomenon at that time. And since 1987 to 2012, scientists work a lot. And why this mysterious sequence present in the bacteria and archaeal genome. And they discovered that it is, a system, adaptive immune system in the bacteria, because bacteria are prone to infected by virus and plasmid, and virus cannot replicate their DNA and RNA themselves, so they need a host. For example, COVID-19 virus use our transcription and translation machinery to make its own copy. So virus when infect the bacteria, actually bacteria, here you can see CRISPR array and also the spacer, and when virus infect the bacteria, through the evolutionary process of the bacteria which survived, they cut a small piece of DNA from the bacteria virus, invading virus, and put into their genome, so that they can recognize same virus when they infect by associated with the Cas protein, that is CRISPR RNA translated protein, and it is like a molecular scissor and cut the known DNA, because it contains the memory. So in the sequence, bacterial sequence, lots of such sequence are present there. So it can cut the virus and make them ineffective and cannot allow them to infect again and use their transcription and translation machinery for making copy. So CRISPR-Cas is a naturally derived molecular scissor, and scientists, especially Jennifer and Emmanuel, when they fully understood the CRISPR-Cas system in bacteria, and then they thought that whether if we change the RNA sequence associated with the Cas protein, whether we can use it as a molecular scissor to cut the DNA of other organism as we desire very precisely. That was the starting point of out of the box thinking, and they practically were successful and published the paper in Science in 2012. So adaptive immune system of bacteria and archaea are now a powerful molecular tool for editing the genome of any organism, living organism, by a precise design. For example, how can it does? This is a simple diagram. You can see here Cas9, and you can design a guide RNA considering the target sequence of the gene you want to edit, and then this guide RNA and Cas guide RNA, find the specific location in the DNA, and Cas9 or any other Cas system can cut the double strand of the DNA. And this double strand break, once it happens, then DNA has own internal repairing mechanism to repair this cut, this double strand break. And there is two pathway already scientists discovered. One is non-homologous end joining repair pathway. Another is homology directed recombination pathway. So naturally what happened, if any damage taken place in the DNA of any living organism due to any carcinogen or radiation, or even by the CRISPR-Cas system, immediately DNA repair it, and during repair some mutation taken place. That means some nucleotide removed the DNA repair mechanism, not exactly the previous DNA, so it has a small indel mutation. For example, one nucleotide is changed, and if you design double guide RNA, you can cut different places, you can delete a gene, and or if you put a donor DNA in the cut position, you can insert a desired DNA in the DNA sequence of your target organism. Similarly, in case of homology directed recombination, after a double strand break, you can put desired the donor DNA with ends homologous to the this double strand, so that it can fill up and gene insertion or replacement can be taken place, because you can change the sequence, only keep the end homologous to the cut DNA. So this is the way you can edit targeted DNA very precisely. And Sophien, as I mentioned, he was one of the champion in application of CRISPR-Cas method for the first time in 2013, because 2012 the first paper, the method was published in Science, and he published in Nature Biotechnology. Two other groups also published plant genome editing, their result in the Nature Biology in the same issue. There are lots of diversities after that discovery, lots of diversities, as well as technological advancement have been taken place in the genome editing by the use of CRISPR-Cas technology. Here I took the image from Sophien's one review article. Here you can see how it does very nice illustration. So a single indel single mutation can be taken place, a classical one, and you can cut designing two guide RNA. You can excise the fragment of DNA and remove it from the DNA, or if you put a donor DNA, you can add it. So lots of options are now we have available. Donor repair template. That means if you want to change the DNA sequence, so what you can do by donor repair template in homologous end joining, you can do like that way, as illustrated here. Dr. Kutubuddin Mullah, he has been working on editing the base. So single base editing can also be possible. For example, CG can be changed into TA. So by using cytidinase, deaminase with NCAS, Nick's Cas9, fusion with guide RNA design. You can do it even multiple base you can edit, and lots of advancement have been taken place. You can do much more precise multinucleotide deletion can be also possible without double strand break by using the prime editing. Prime editing is the most advanced one, so you can do more precisely, more accurately, more faster the gene editing, and you can do it in any other cell. In case of plant, stacking beneficial alleles, for example, by using CRISPR-Cas homology directed recombination, diffused beneficial alleles, you can edit and you can stack beneficial alleles in the chromosome. Here you can see. So it can also be done for making a robust change. So these are the principles. Another issue is associated with the CRISPR-Cas genome editing technology application in plant cell is how can we deliver the CRISPR-Cas reagents inside the plant cell, because plant cell cell wall is a very thick cellulose containing structure, and in that case it is a bit challenging. There are several methodical advancement already been developed by different scientists. For example, biolistic bombardment, nanoparticles, or agrobacterium, classical one, tumefaciens mediated one, polyethylene glycol, or antiviral RNA. Through these processes, you can inject the CRISPR-Cas system into the interior of the cell for the genome editing. So it has significantly been advanced. And in the most of the journal, in the method, protocols are written in very briefly. So if any essential step is missing, then it is difficult to reproduce. This is why we took an initiative to publish a book, CRISPR-Cas methods. And first issue is now online, and second issue is now on the pipeline. So CRISPR-Cas, actually first CRISPR, at that time it was not a CRISPR. It was just a strange, segment of DNA repeatedly observed in the chromosome, in the DNA genome of a bacterium by Japanese scientists. Until now, it traveled a lot. And from 1987 to 2012, scientists made a big breakthrough, basic research translated into a technology, CRISPR-Cas9 technology by Jennifer and Emmanuel and some other groups also. And later, it has significantly, incredibly faster moved to the practical application. This year Nobel Prize recognized that such a fast-moving subject. And CRISPR is obviously unique because it derived from a natural process found in bacteria to protect themselves from the pathogens like fuzz or plasmid, targets genes for editing or regulating, and it is comparable to Photoshop. And CRISPR technology is superior because previously there were some genome editing technology just like zinc finger nuclease or TALEN for site-directed genome editing, but they were proven as cumbersome because they require sophisticated protein digestion, synthesis, validation. But in contrast, compared to the earlier genome editing technology, CRISPR is cheap, relies on single guide RNAs to target site specificity, can be readily designed based on genome of choices in multiplexed fashion, in which several genomic loci can be perturbed or edited at the same time through multiplex genome editing. System creates stable and heritable mutation, transgene-free plants in a few generations, because transgene is a big problem, biosafety issue, and it takes a very long time to practically apply the technology. But in case of CRISPR, you can get the mutant, classical mutagenesis we use. So transgene-free plants we can get within few generations. Edited plant variety can be used for plant breeding because you can make huge genetic diversity by chopping or editing the genome of a particular plant and make a huge genetic diversity for plant breeding. New set of CRISPR-Cas systems such as Cas12, Cas13, they are much more convenient. So future of CRISPR-Cas, it is, Jennifer and Emmanuel, they propose like that way. It can be used in human gene therapy, agriculture, crops, animal, synthetic biology, pathway of engineering, programmable RNA targeting, screening for drug target, ecological vector control like mosquito sterilization, viral gene disruption, pathogen gene disruption, diagnosis of the pathogen, and much more, because it is a powerful bioediting. We can rewrite the life by using CRISPR. Outbreak of rice blast, as I mentioned, it is a very big threat, and nearly 15 to 20% of the total rice produced all over the world are damaged by this one. And scientists already applied CRISPR-Cas technology for mutagenesis of transcription factor gene OSERF922 in 2016, and they found blast resistance. And the gene editing is not only that we are doing in the laboratory. In naturally edited gene already been existed, and lots of scientific paper describe it. For example, semi-dwarf SD1 green revolution rice contains a defective gibberellin 20 oxidase gene. That means naturally a defective gene. So now by using CRISPR-Cas, we can make it, manually, and it should be as it is naturally being exist. So we can, apply it more faster to the practical field. For example, BDH2 gene is mutated in the aromatic rice. So aromatic rice, they have BDH2 gene mutated. So in our high yielding variety, if we simply 8 base pair deletion in exon 7, if you do by using CRISPR, you can make our high yielding variety into the aromatic, because most of the aromatic rice indigenous varieties, they are very poor yielding and susceptible to very many diseases. In our lab, we have started this project. And there are lots of success stories in application of CRISPR-Cas technology in, gene editing for changing different traits. For example, one of the classical and success story is the editing of mildew resistance locus, MLO, in tomato. You see, just deletion of this 48 to 49 base pair resulted in immune plant, and in the transformant there is no trace of tDNA or any other transgene. And CRISPR-Cas technology for improvement of crop yield, because it is a polygenic character and very difficult. In China, by using CRISPR technology, they edit the genome of rice. The team of Ying Wang from Syngenta Biotechnology China designed several CRISPR single guide RNAs and successfully deleted fragments of the dense and erect panicle DEP1 gene in the indica rice line and improved which the transformant or edited plant is improved in yield related traits like dense and erect panicles, reduced plant height, and these are the examples. And by using base editing, several base editing technology already applied in rice, one of the major food crop, and the result is nutritional improvement, herbicide resistance, enhanced nitrogen use efficiency, because we spend huge money for feeding the plant, urea and other nitrogenous fertilizer. So nitrogen use efficiency, if we can increase by using gene editing, we can save huge money and obviously reduce the environmental pollution, regulates cell senescence and death. So these are the success stories. Lots of success stories are available. One of the fascinating success story is the domestication of wild tomatoes, to the the modern one, and through the evolutionary process, breeding process over 10,000 years, wild tomato changed into the highly productive our cultivated tomato. Scientists identified what are the genes domesticated, and they introduced all the genes into the genome of the wild tomato. And they found that, surprisingly, the edited plant is just like the domesticated one. In addition, they found that the traits, retention, biotic and abiotic stress tolerance, the properties in the wild, counterparts, that is wild tomato, retain here, which is very, very useful for, fighting against the climate change. So this is a new area. Multiplex genome editing technology can use wild or neglected, or orphan crop plants to domesticated into the de novo process. There are lots of other examples for, editing plants to improve resistance against the pest pathogens, as well as against the different abiotic stresses in many plants. I am not going one by one because I am excited to listen the talk from Sophien. Now you may be asked a question: is it still only in the laboratory, the CRISPR-Cas genome editing in the plant? No, it has been progressed significantly, and some of the products are now in the field. For example, soybean, disruption of DRB2A and DRB2B genes for drought and salt tolerance edited in soybean, and it received the green pass from the USDA for practical application in USA. Similarly, camelina, which is a oil producing crop, and editing of undisclosed gene is not disclosed for increased oil content. It is also released in the practical field. Several others, similarly, waxycorn, very high content of amylopectin, which is achieved by inactivating endogenous waxy gene, WX1, that encodes a granule-bound starch synthase catalyzing production of amylose. So DuPont pioneer in USA, they developed, gene-edited waxycorn, and it has already been released in the field. And Dr. Kutubuddin Mullah, who is now panel discussant, his supervisor at Pennsylvania State University developed anti-browning, white button mushroom. When you cut it, browning is a problem. So anti-browning mushroom, by knocking out a gene associated with the phenol oxidase, polyphenol oxidase, resulted no browning, and it is also received the green pass for cultivation. That means CRISPR-Cas gene-edited product are now in the real life. And in our case, in Bangladesh, I really excited from the proposal from Sophien Kamoun to use CRISPR-Cas gene editing technology to fight against a new enemy, wheat blast, which devastated 15,000 hectares of wheat in Bangladesh in 2016. Yield loss was up to 100%, and it is a terrible disease which has been in South America since 1985, but other continents are free. And in collaboration with Sophien Kamoun, we first, within weeks, by using field pathogenomics, we, determine the genetic identity and origin of the wheat blast. Here you can see 31 researchers from four continents participated, and it was actually, the idea was a brainchild of Professor Sophien, who will deliver a talk very soon. And this paper already cited 181 times, and it was a big news because without any funding, without any project, how international scientists, work in a concerted manner, and we were able to, Sophien's leadership is amazing. I received a fantastic support from him to organize all these people to make this breakthrough within weeks. And then we decided, how can we solve this problem? As you know, S genes are negative regulator in the disease incidence, and if we can make dysfunctional of the S gene, then disease resistance developed, as I have shown in case of tomato powdery mildew disease. So we targeted several S genes orthologous to rice and to edit them. And fortunately, Sophien, Nick, and Emma Wellington, we collaborated, and Sophien also and Nick visited Bangladesh to really see what is wheat blast and how farmers are frustrated due to this dangerous disease. And he heavily engaged in this project, and he trained our people on genome editing technology at Bangabandhu Agriculture University. 50 young scientists we invited from different institutes to receive the training. And he also invited 2 members from my team, and they were Mahmoud and Sanjoy. They worked 4 months in his lab, and they received fantastic training. And we also received a very nice international collaboration from Pankaj Bhaumik, as he developed a very nice tool for transformation of wheat, because wheat is a hexaploid crop, and editing of wheat is or genetic engineering of wheat is very complicated. Pankaj developed a protocol, and he helped us to edit the wheat some S genes. And through collaboration with Sophien and Nick and Emma Wellington, we proved the concept that gene editing in wheat wheat is possible as gene editing, and our target is to apply this concept to our local varieties, and we are now testing the resistance of the blast resistance of the edited plants. Unfortunately, in the laboratory, we found some moderately resistant, not like a dramatic resistance in case of powdery mildew in the tomato. So that idea, when we participated in an international competition, we proposed that genome editing of wheat could be useful for developing blast-resistant wheat variety in Bangladesh. That idea contest, our team was champion and received Islamic Development Bank Prize in 2018. And I would like to share with you the last topic that is a fascinating topic, the CRISPR-Cas, that is Cas12 enzyme mediated new technology, because in case of wheat blast, we had a very big problem of precise molecular diagnostic protocol was not available, and the protocol discovered by other scientists, we found them, they are not precise and accurate. And in collaboration with Ohio State University Professor Guliang Wang, USDS scientist, and Chinese Academy of Scientists, we were able to find two segments of DNA in the wheat blast genome. First one encodes a putative acetyl protein. Second one encodes a DUF341 family protein. They are unique and not present in any other blast fungus. So we designed primer, and we found that it is very specific for diagnosis of wheat blast. But PCR, we cannot make it affordable and available in the quarantine system as well as in the farmers level. This is why what we did, we published this paper in Engineering recently during coronavirus. We used the Cas12a and guide RNA, because as we know, the specific sequence of the in the DNA of wheat blast fungus. So we designed a protocol to make a very convenient and rapid technique to detect the wheat blast. Sherlock method developed by Feng Zhang for detection of coronavirus. Similar method we used because guide RNA can detect that a DNA sequence, and Cas12 enzyme can cause hydrolysis. And here you can see this strip, if you put into the infected sample in the buffer solution, and then Cas12 enzyme cut the that DNA and cause lysis, and you cannot find the band, and it is the positive. So in case of others, they are negative or uncertain. So this, as wheat is #1 exporting and importing food crop, so it can be used in the quarantine all over the world as well as in the farmers field, and we can now detect wheat blast within 30 minutes. We validated this method in the field samples also. So this is an amazing application of CRISPR-Cas system in addressing detecting a pathogen, and it can be used to detect any other organisms. So I'm at the end of my talk. CRISPR-Cas mutants basically indistinguishable from spontaneous mutants or from mutants induced by chemical or radiation when lacking the transgene construct. So in this case, it should not be required to pass through all the strict biosafety guidelines. But in case of Europe, still plant products from mutagenesis specifically exempted from EU GM regulation if it is done by chemical mutagenesis, which is not specific, or radiation. But in case of CRISPR-Cas, they do not consider it as the mutagenesis this product, he has to follow the GM regulation. In many crops, cultivars from mutagenesis, wheat, and others now worldwide cultivated even in Bangladesh without any biosafety, because BINA, Bangladesh Institute of Nuclear Agriculture, they're using the gamma radiation, and lots of varieties they discovered, and they are now in the field. But if you do precisely a single mutagenesis, and now if it is required to pass through a long biosafety procedure, then it is problematic. Transgene-free CRISPR-edited crops are outside of GM regulation in Japan. Japan is a very critical country, you know, a very stringent their system, but even though they realize that transgene-free CRISPR-edited crop should get the, you know, direct practical application. And US, Canada, they also approved. But other countries, they are waiting. In Bangladesh, we have no law yet been formulated. I am a National Core Committee member of crop biotechnology. I am advocating to make a law like Japan or USA. I would like to show you a recent publication. Here you see in Nature, Fusarium head blight is a big problem. It is published in May 2020. Here you see from a fungus, a gene, FHB, through horizontal transfer moved to a germplasm of wheat. And if you use this germplasm in classical breeding and transfer the gene to the high-yielding variety, then it is non-GM, exempt from the regulation. But if you clone the gene and if you introduce it very precisely by using gene editing or agrobacterium-mediated transformation, then it is GM. And this is really a big problem, but this gene is very, very useful in reducing the, mycotoxin as well as the control of Fusarium head blight in the wheat, and that one is also a big disease. So my concluding remark is the world population is expected to increase about 10 billion by the year 2050. The CRISPR-Cas system became a gold standard revolutionary method for breeding smart crop for resiliency in climate change. And CRISPR-Cas toolkit, I want to say it toolkit because it is simple, efficient, a versatile revolutionary technology for investigating plant biology. It would be a very powerful, and it is a very powerful technology to dissect and understand the, mystery of life, not only plant life, as well as doing precision breeding and creating genetic diversity in plants. We foresee that the application of CRISPR-Cas technology in various crops revolutionize agriculture in a new green revolution to ensure food and nutritional security of the ever-increasing world population. And I expect that this technology would be a big, impact in our biology, and every laboratory like PCR, in future we shall have this technology in our hand. But what we need to do in our curricula, especially in university biology course, as well as under our school level, high school level, we need to introduce this technology to make, to generate young generation future scientists to handle this technology and make big impact. I am very much thankful to Professor Suhail Abdullah, Science Porter Bangladesh, and other team members, young team members who are, organizing series of these fantastic webinar. And I am also thankful to Sophien Kamoun, Nicholas Talbot, Pankaj Bhumi, Guliang Wang, and my students, team members, lots of international collaborators, including Kutubuddin Mullah, and I am also thankful to lots of donors who have been supporting tremendously for establishing my dream institute of biotechnology and genetic engineering at BSMRU. We have now a fantastic facilities at our university. So if any young minds have new idea to do, use Frontiers technology to address any challenges in agriculture, in microbiology, you are most welcome. Thank you so much.
Thank you, Dr. Tofazzal, sir, for such a wonderful presentation, I should say. It was very captivating, and I learned a lot from you, sir. We're lucky to have you with us. And now I would like to request Dr. Sophien Kamoun, Senior Scientist at the Science Bar Laboratory in the UK, and also fellow of Royal Society. So Dr. Kamoun, I will request you to power up your presentation, sir.
Thank you very much. And thank you, Tofazzal, for your wonderful presentation and your enthusiasm, really, more than anything, because essentially what we really need with new technology is enthusiastic people like yourself to really promote it and make sure that this technology is reaching the people who need it the most. Because I think the message here, more than anything, is that technology can be game-changing. That's correct. We know that. But more importantly, technology can really change lives. And I think we know that when we look around ourselves and see the world we live in, we can see the impact of technology. And this is the technology of the future. As you mentioned, PCR now became a household name because it's being used for detecting this terrible coronavirus. And that's a new tool for molecular biology, and CRISPR is the type of tool that would have the impact, is already having the sort of impact that PCR has. So thank you again for your wonderful presentation and inspiring words. I suppose everybody can see my slides, right? Just want to be sure that...
Yes, sir. Yes, sir.
That's happening. Okay, excellent. Super. So I think we've heard a lot about CRISPR already, so I'm going to maybe skip a little bit faster over some of the slides, but we even, in the last years, really, CRISPR became a household name, as you know, and we even had this terrible incident with CRISPR babies. I hope we don't hear more about that in the near future, but what we're really interested in is the impact on plants and plant technology and plant breeding. And I want to maybe give you a slightly different perspective than Tofazzal, maybe complementary to what he presented. So I'm going to take you through the historical developments that led to CRISPR. So we should first realize that CRISPR, more than anything in plant biology, is a reverse genetic tool. So I know not everyone here is a biologist or geneticist, so let me briefly describe what reverse genetics is. It's basically going from a gene sequence to a mutant. And to do that, we need methods. And some of these methods are actually available, and they were available back in 2012 before CRISPR was invented. You can see some of that here. We can use random mutagenesis. We can use approaches like tilling. We can knock out genes with agrobacterium tDNA or transposons, and so on and so forth. We even had these site-specific nucleases like the zinc finger and the talons. What CRISPR really did more than anything, it just made this approach of reverse genetics very easy to implement, fairly simple to implement to any genes, any sequence. And also it really opened up the technology to a lot of different plants that are not necessarily model plants, whereas some of this technology are limited to model plants or crop plants where there are a lot of resources. And the technology is really centered on these so-called site-specific nucleases. So for those of you who are really early career students, if there's something you want to read about, really is the site-specific nucleases. And before CRISPR, we had, before Christmas, I was going to say, CRISPR, zinc finger nucleases, tile effector nucleases were available. These also basically cleave DNA at specific positions. They are programmable, but programming them is difficult because you have to basically make a new protein every time you want to target new sequence. And that's tedious, expensive, and slow, and not always very precise as with the zinc finger nucleases. So CRISPR came because it was easy, because the specificity of where the scissors are going to cut in the genome is not provided by the protein, by the Cas9 protein or other Cas proteins these days, but by an RNA, the so-called small guide RNA or sgRNA. And this RNA is the sequence, carries the sequence that provides the specificity of where this enzyme is going to cut in the genome. And that made things a lot simpler because you just don't need to change the enzyme. You just have to change this guide RNA. So as you heard already from Tofazzal and also from the recent news about the Nobel Prize and so on, there's essentially CRISPR Craze that started in 2012, really with this paper of Emmanuel Charpentier and Jennifer Doudna, who basically showed that Cas9 cleavage of a particular sequence can be programmed with a particular guide RNA. In this case, they made a synthetic guide RNA, which is a fusion between the two natural guide RNAs of this enzyme, of this Cas9 enzyme. And that was published in, summer 2012. I remember vividly when I read that paper in August 2012. And it influenced a lot of us to really ask the question, can we apply this programmable system to create cuts in the genome of the systems we study, of the organisms we study? And it didn't take very long. It was really early January 2013 before the first examples, the first papers of using the CRISPR system, CRISPR-Cas9 system to essentially cause targeted mutations in human genomes, in this case, were published. And then there was a whole flurry of papers basically showing that this system can be used in pretty much any organism, including plants, as Tofazzal mentioned. There was actually 3 papers that were published in Nature Biotech, but there was also additional paper around the same time that came out. And so that CRISPR craze really started infecting plant scientists in a year after the Doudna-Charpentier paper around in summer 2013. So what can we do with CRISPR implants? I know that this is a broad audience, so I don't want to get too technical, but I decided that I should highlight some of the articles for the students who want to learn more. So I really encourage you to read this wonderful article by one of the leaders of the field, by Kaisha Gao and her. Kaisha is a pioneer of gene editing before the CRISPR system, and runs a very active and dynamic research group in the Chinese Academy of Science in Beijing. And I really recommend her work and her papers. But here it basically summarizes this figure from her review article, summarizes the different applications of the CRISPR technology to plant biology. So we can knock out genes. This is the easiest thing we can do. We can basically target a gene in the genome, any gene, and basically take it out of action, if you like, by deleting it or creating mutations that are make that gene non-functional. Gene knock-in is another application of basically replacing a sequence by another sequence. For example, replacing a gene by another gene, or by a stack of genes, as this cartoon shows, and or perhaps doing a correction of a gene. If your gene carries a bad mutation, you can actually fix it. And so on and so forth. I think I don't want to go through everything, but I think Tofazzal also covered some of that. But the key, really, the simplest and most straightforward application is a gene knockout. You're basically taking one gene out of action. And I'll just show you some of our work. Some of these date back to the early days, where it's fairly straightforward to make small deletions, for example. All you need to do is to make 2 guide RNAs now, not just one, as shown in this cartoon. So you can have a small deletion of 70 base pairs if you use guide RNA 1 and 2, or a larger deletion of 400 base pairs if you use guide RNA 1 and 3. And so by delivering these guide RNAs with the Cas9 enzyme in the plant cell, you can create deletions, and they can be easily identified by PCR because you have smaller fragments that will amplify. And then, as you can see here in the cartoons, by sequencing, you can see that the top sequence is the wild type sequence, the normal sequence of this plant, and then below that you have these various deletion mutations of about 70, 75 base pair deletions. It's also possible to make big deletions, even very big ones. So what I showed you there was a deletion of maybe tens or hundreds of base pairs, but we can make actually deletions of thousands of base pairs or tens of thousands of base pairs. This particular example is a deletion we made in tomato. of 53 kb, so it's 53,000 base pair, and four genes actually that were deleted in one go, basically, with a series of 4 guide RNAs that were targeting similar sequences in these paralogous genes or related genes in this one gene cluster. This happens at extremely low frequency, and we were fortunate, in fact, to recover this particular mutant. But it does happen. We still don't know exactly how to improve the frequency of getting these big deletions, but if you generate enough transformants and screen enough, you might be fortunate to identify some of these big deletions, which can be very useful because you'll be taking out of action multiple genes in one go. Okay, so now I'm sure all of you are wondering, how do we do this? How do we deliver this CRISPR technology implants? And also, especially in Bangladesh, in India, and any other countries, there's a lot of interest in applying this technology. So I thought I'll have a slide here on basically the steps required to perform CRISPR from A to Z and perform CRISPR implants. And what this slide shows is really the 4 steps that are necessary to perform CRISPR. And as you can see, there are 4 also different types of expertise that are required. So this is usually done as part of the teamwork. And so you need bioinformatics first, because you need to design these guide RNAs and design also cloning strategy for these guide RNAs. And it's possible to actually compile these guide RNAs in one particular cloning vector, so in one construct, if you like, and this can be even 10 or even more guide RNAs in one particular construct. But this requires design, this requires computational biology, this requires some bioinformatics skills. The next step is the molecular biology per se, which is the cloning and of the plant transformation vectors. This often includes having the Cas9 and the guide RNAs in one particular plasmid on one particular vector. This is also requires some skills, and there are some in the workshops we give. We do talk a lot about, for example, the Golden Gate method of cloning, which is really important in enabling, for example, concatenating multiple guide RNAs in one particular vector. Step 3 requires, again, totally different type of expertise, because step 3 is about plant transformation, and that often requires skills and access also to the right facilities and equipment to perform plant tissue culture and plant transformation. And in my view, this step is actually often one of the most difficult one to bring to a lab, because it does require fairly heavy investment in terms of facilities and in terms of clean rooms and so on and so forth. Also, in many cases, for many crops, especially the orphan crops, the crops that are not very well studied, and maybe some of these emerging crops, we don't actually have methods and protocols for plant transformation, for transforming them. So I do feel that plant transformation, tissue culture is really an area where we need to make a lot of development, especially as targeted to developing countries. Also, in other cases, that the crop may be transformable, but the varieties of interest in a given country may not be transformable, and you do want to introduce your mutations in that particular variety of interest, a local variety that is already adapted to the conditions of the country. And finally, the last step is genotyping, where you want to identify the mutations you introduced. And the genotyping can be done by PCR, but can also be done by sequencing. And this also can be tricky if you're talking, for example, about plants like wheat, where you have 6 basically copies of every gene, because wheat is a hexaploid. And then on top of that, many of the genes you might be interested in could be present in multiple copies. So it becomes quite complicated to genotype and so on. The one step I didn't include here is phenotyping, but I think in general, there is, in most plant breeding facilities, there is the expertise to phenotype. So I'm not so concerned about that last step, really, which is the phenotyping, identifying basically plants with valuable traits and valuable mutations. I think that usually is available in plant breeding institutions. Okay, so this is again a slide from a figure from Kaisha Gao's review where they summarize the different methods of plant transformation. And this actually gives you an idea of the fact that there are multiple technologies. This can be done through particle bombardment, for example, or through agrobacterium mediated transformation. And this can be done with the Cas9 enzyme already in complex with the guide RNA, or it can be done with a fragment that is expressing all these this system. So there are multiple variations on this, but again, I do feel that this particular step of plant transformation deserves a lot of attention and a lot of investment. I feel that that's really the weak point in delivering this technology to many places. There is a very exciting paper that just came out really literally weeks ago from our colleagues at UC Davis, Jörg Dubkowski's lab. And in this paper, what they did is they generated an engineered transcription factor, something they call the GRF-GIF. This is a growth factor, essentially plant growth factor. And they showed that when they combine this GRF-GIF chimeric protein with transformation, they have massive increase in transformation, in regeneration frequency of plants after transformation. In particular, in wheat, this has a massive impact. And this is really applicable to the CRISPR technology because this can be combined, this construct can be combined together with the CRISPR system and therefore at the same time provide the gene editing, but at the same time also increase the frequency of regeneration of transgenic plants. And I think this is really important because this is again addressing that weak link in the CRISPR pipeline, and hopefully this technology will have a wide impact both on engineering bioediting of wheat, but also of other crops, and especially recalcitrant cultivars adapted to local environments in developing countries. Okay, so now I'm gonna switch gears and talk a little bit about the applications of CRISPR to agriculture. Again, we heard a lot of that from Tofazzal, so I don't need to go through all of these. But you can see here on this list really the very obvious applications. We talked also about de novo domestication earlier. Tofazzal mentioned that this is one of the kind of exciting, completely sort of new approach to really plant breeding that CRISPR enables that wasn't really accessible before the CRISPR technology. But there are, of course, applications to plant health, to drought, abiotic stress, etc., improving nutritional value, even removal of allergens and toxins, making the food essentially more palatable and more nutritional. And with less allergic reactions, which is something that is increasing as we move to more industrial type food systems. And this figure here it really illustrates the advantages of CRISPR compared to more classical approaches. I mean, again, I'm always reluctant to present CRISPR as the only tool in the toolbox. There are many other tools that are available, and those of you who are involved in plant breeding, in mutagenesis breeding, and so on, and also transgenics, should not feel that CRISPR is really replacing this. This is an alternative to these existing methods, but it is an alternative that has many benefits, and one of them is actually speed, and it is faster method to really deliver a particular trait in a variety of interests. So again, I encourage you to go through this particular review article. There are also some very creative applications of CRISPR, especially now in countries where gene editing is deregulated. This is the case in the US, for example, and this is work by our colleague, Zach Lipman at the Cold Spring Harbor Lab in upstate New York. And what Zach did is he created this triple mutant. So this is a triple mutant in tomato. 3 different genes are mutated in this variety called Sweet 100. And this variety is, as you can see, extremely compact, extremely bushy, and is basically adapted to urban gardening. So the Sweet 100 is very compact, and it's really perfect for places where you have very little space. And even if you think about in your balcony, it could be just perfect tomato to basically maximize yield with versus space. So this is, we think about this technology having been invented only in 2013, and we already have directly a variety that can be used in urban gardening and so on. It's just so exciting to think what will happen in the next 10, 20, 30 years. I mean, this is just 3 mutations in one tomato variety, and look how interesting this particular variety looks like. So we can think about how this method really enriches our creative potential to generate plants adapted to all kind of environments.
So, Dr. Kamoun, I'd like to, Dr. Kamoun, I'd like to I'd like to interrupt you. Your slides are not visible, so I'd request you to stop the presentation and then power up again.
Okay, I'm not sure what happened, but let me do that. Resume presenting.
Yes.
Yeah, could you see the tomato?
Yes,
the urban,
yes.
Yep.
You'll have to go. Yeah, you can see it. Yes, sir. Continue.
Okay, perfect. Yeah, I'm not sure what happened, but thank you for alerting me. So this is if you missed it earlier, this is the Sweet 100 tomato that is adapted for urban gardening. It has issues, by the way, and I'll tell you in a second what the issue is. So, so application to plant health. Obviously, I'm a plant pathologist, just like Tofazzal, so our main interest is in making plants more resistant to pathogens. And here the concept that is important to know is the concept of S genes, or susceptibility genes. And the idea here is really through loss of function or change of function, we can make disease resistance. It's not about bringing new genes, as in resistance genes. It's really about perturbing some of the existing genes that are important for a pathogen to establish itself and cause disease. And this is the concept, and I point to this review article if you're interested in getting more information about this topic, and for the students who want to study more on this, I really encourage you to read this article. But the idea here is this is a cartoon of our understanding of plant pathogen interactions. So this is the blast fungus here developing effector proteins in the host cells, and these effector proteins have targets. And these effector targets are essentially host proteins, host molecules that are important for the pathogen to establish itself. So if you take those out, the pathogen will struggle to cause disease. It's almost as if you take, if the pathogen has a key and you take the key lock, or you take the key hole from the door, then the key would not work anymore, and then essentially the pathogen will struggle to really enter through that door. So that's one type of essentially of S gene, can be the product of the targets of these pathogen effectors. Another type of S genes are essentially plant genes that are negatively regulating immunity. So these are, the plant doesn't want to have the immune response on all the time, because that's basically like when we have a fever, we are not functioning very well. So the plant also, when it has an immune response, it's not functioning very well. So it has all kind of regulators, negative regulators of immunity. And if you can mutate those negative regulators of immunity, will increase immunity. And that can, in some cases, under some circumstances, that could actually yield a beneficial phenotype. And our understanding of the negative regulation of immunity is actually quite complex and quite not yet fully developed. So there's a lot of potential also for exploiting these negative regulators. When I present this, many people ask me the question of, well, how can you really make a plant better if you remove a gene? I mean, that gene is there for a reason, right? Many people struggle with that concept. They just ask me like, why you can't? they can't work because you're removing a gene that's there. So that gene evolved for a reason, it's there for a certain function. And so my answer to that question, which maybe some of you are wondering about right now, is to show you this picture of this very ugly creature. So this is a domestic cat. It's a breed that is hairless. And it has actually mutation in the genes that produce hair. And the reason I'm showing you this is because the crops we grow are not wild plants. They are grown just a little bit like domestic cats. They are pampered in our agricultural fields. And so those genes that we're talking about, some of them, they are important in wild systems. When the plant is growing in the wild, that's where it evolved. So just like the hair of the cat, the hair of the cat is very important for the wild cats. This cat would not do very well as a wild cat. But if they're grown in our pampered environments, whether it's your house with this cat or whether it's an agricultural field, then some of these genes become actually dispensable. We could remove them and still have a crop that is properly functioning. To give you an example, let's say the genes that are involved in flowering in potato, they're really not contributing much to the production of potato tubers. So we could mutate potentially genes that are important for potato flowering, and that will not impact the production of potatoes in your field. That really wouldn't matter, just the same way as this ugly cat here doesn't really need the hair to survive in someone's house. Okay, so now we want to target S genes, and how do we do that? So this is tricky because this requires basic fundamental research. Often we need to have some degree of understanding of the disease mechanisms and pathogenicity mechanism to really know which genes we need to target. But one approach sometimes that can work or may not work is to exploit knowledge known in one area in one plant, in one system, to another system. So in this case, this is what we did for this MLO project. This is a powdery mildew susceptibility gene, where a knockout mutation in these genes gives resistance to powdery mildew. And this was known in barley. So this was actually described in barley. And what we did is we identified the homologous genes in tomato that was similar to this MLO gene in barley and introduced this mutation in that gene. And that resulted in the tomato that is resistant to powdery mildew. So that's an example of how we can transfer the knowledge from one system to another, because in many systems, in many important crops pathogen systems, we don't actually have enough basic knowledge, or that's a little bit slow to generate really information about which genes to target. So that was our proof of concept with tomato, and this led to this project that we received a few years ago with my colleague Nick Talbot, Emma Wallington, but also with critical collaboration with Tofazzal and his team. And the idea here was to do the same as we did with Tomelo, because we don't have a lot of information about the wheat blast disease. It's a fairly new disease. There's not a lot of basic research on its molecular mechanisms, but there's a lot of research on the rice blast system. So what we did is we took genes that are known in rice to mediate susceptibility to be S genes against the rice blast fungus, and we identified their orthologs in wheat, and then we targeted these genes, about a dozen of them, in wheat with the CRISPR system to mutate them and create mutant populations of these genes. This project actually really ended up being much larger than I expected when we designed it, because we ended up with about 6,000 different lines, transformant lines of wheat, and this is just the first generation. So that's a lot of plants to really deal with. So the screening, a lot of it is still in progress. But we did a number of proof of concept studies. One of them was in barley initially, because this is a system that is easier to transform, and also with a little bit more knowledge than wheat at the time, at least in terms of the genome. And we could show that we can actually deliver this CRISPR system in a cereal crop and also target the gene important for susceptibility to the blast fungus. And this is actually infections with the Bangladeshi strain or lineage of wheat blast on these mutant barley plants. And as you can see, they look quite promising. And this is the gene we targeted here, is the so-called HMA gene. It's a gene that is known to be an effector target and known in rice to be important for susceptibility to the rice blast fungus. So we went on and did the same thing in wheat. In wheat is much more complicated. First of all, we have the three different genomes. So wheat is a hexaploid. So we have A, B, and D genomes. And in addition to that, a lot of gene families are expanded. So this HMA family, for example, in wheat is much more complex than it is in barley and other plants. And as you can see here, there are many copies, up to 7 copies in the A genome of this HMA gene. There's five copies in the B genomes and 4 copies in the D genome. So it's really tricky to know which genes to target. And it's also really tricky to know whether your mutants have actually have mutations in all of these genes or not, which may be essential for the system to work. So wheat is a lot more complicated. But as you can see here with the cartoon showing the scissors, we decided to target as many genes as possible with as few guide RNAs as possible. And then the genotyping is also complicated because now we have also a system where we have multiple copies of these genes. How do we genotype them? This wasn't actually simple because a lot of these genes are also related, so it's not easy to design primers that are specific to one region or another. And what we did here is we essentially decided to use sequencing, amplicon sequencing, where we actually amplify different fragments that overlap the regions we are targeting. So this can be the green bars here or the yellow bars, and then mix them all together and then do genome sequencing for each one of the mutants we have, and then determine whether we have mutations or not. So it's been really difficult to precisely genotype the mutants we have, but we could see that we have mutations. So we have been successful in introducing mutations in wheat. This is an example, a very straightforward example where we have 2 guide RNAs illustrated by those red scissors, which create this particular deletion. And you can see here from the sequences we recovered from that amplicon sequencing method that we did introduce this deletion in this particular variety. This is a case where it looks like we have a much larger deletion because the amplicon sequencing basically revealed the bigger deletion. We don't even have the sequences matching that guide RNA on the right side. And then this is a case where, again, it illustrates the complexity of wheat being a hexaploid, is that we have now a mixture of deletions that are targeted to that region and then bigger deletions. And it's really difficult, again, to sort them out, especially with the complex gene cluster like the HMA cluster. Nonetheless, we have, as you heard also from Tofazzal, we have completed now the screen of the 6,000 original lines and also these HMA-targeted mutants. We have some promising phenotypes, but we are still validating them. And I should also highlight that we got fantastic help from Mahmoud and Sanjoy, who came to visit the lab during that period and contributed tremendously to this screen and also to other projects in the lab. So we now are planning the next phase of screening the next generation of the most promising mutants we recovered, and hopefully we'll have some positive results to report. I should add that we got a bit delayed because of the restrictions due to the virus pandemic. So with that, I think I will finish here, so we have time for discussion. I just want to highlight this brief text that I wrote, actually, thanks to Tofazzal and his co-authors and co-editors who invited me to write the foreword for their recent book on CRISPR-Cas methods. And I want to emphasize this particular paragraph because it really means a lot to me. I really, as a scientist and as someone who grew up in a developing country, I really care a lot about making discoveries, fundamental discoveries, advancing our science, but I also care a lot about bringing new knowledge to developing countries and to the young people who are really the future. I mean, the students now in the audience, you are growing up with this technology. So I mean, I cannot even foresee how things will be in 20, 30 years when you will be in our positions and all the applications that will be possible then. This is a very new technology again. We're talking about just a few years. And the perspectives are just limitless. And so I want to highlight here the statement that my hope is that this book will guide a new generation of plant gene editors from all over the world. I hope it would inspire early career scientists, especially from developing countries like my own country, to embrace gene editing technology and forge their own path in this rapidly expanding area of genetics. So this is the future. So those of you who are now hearing about this technology and really growing up with it, this is a very exciting moment for you to be a biologist and to be a geneticist. And please do consider this technology. As an example, by the way, I want to mention this because when I was a PhD student, PCR was invented. And now it's a household name, isn't it? Because of everybody heard about PCR because that's what's being used for coronavirus diagnostics. So the future is bright, and we're really at the very beginning of the applications of these methodologies. So with that, I want to thank my many colleagues. I want to go back to this work we did back in 2016 and has been the foundation of a wonderful friendship with Tofazzal and with your country, Bangladesh, and many contributions from Bangladeshi local scientists, but also from others around the world who are listed here. And I also want to thank my team, Vlad Nekrasov, who now has his own lab in Northampstead, doing actually gene editing in wheat, is the one who started back in 2013 the application of CRISPR to wheat, and then various members of my lab. In particular, I want to highlight the more recent ones, Diana, Adeline, and Torsten, who were involved in the wheat bioediting project. So thank you again for your attention, and I hope we have time for discussion. I'll stop presenting here. Thank you again.
Thank you, Dr. Sophien Kamoun, for such a captivating presentation. That was a very brilliant presentation, and it was very inspiring as well. You're from a developing countries, as you said, and this will definitely inspire some of us to work with the CRISPR-Cas technology and the tools. Now I'd like to request Dr. Kitibudin Alimullah, a scientist at the National Rice Research Institute of India in Kutchak. Dr. Mullah was the speaker of our first seminar, and I'd like to request Dr. Mullah to share a bit of his research and to talk a little bit about this seminar and to add to this conversation. So Dr. Mullah.
Yeah, thank you, Sham. So it was wonderful from both the presentation. So I think I should go one by one, and you'd request both Professor Islam and Professor Kamoun to switch on their mic because I have some questions for them.
Absolutely. Go ahead and ask.
Okay, so I think my strategy is to go for the presentation of Professor Sophien Kamoun first because we just ended that presentation, and then I go to Professor Islam's presentation. Yeah. So Kamoun, means Professor Kamoun, I was really looking forward to listen to you because we started genome editing much later than you have started back in 2013, and I now focus on base editing technology mainly to alter single nucleotide changes in the genome. And so it was wonderful to listen to you how you started with small deletion using two guides, and you also recovered a 53 kb deletion. That was wonderful, So a whole portion of genome could be deleted with that. So my question on that part, that have you seen any kind of genome replacement or inversion kind of things while you are screening for those mutants?
Thank you again for your question, and the answer is in the particular mutant, the 53 kb mutations. It was not a clean 53 kb mutation. It was actually a combination of two events. First, a very big deletion, and then a smaller rearrangement next to it. So we have published this work. So I encourage you to look it up, and I can actually post the link in the chat. But I think the issue of structural variation is a very important one you're bringing. I think that's one that's difficult to study, and we've all been very focused on SNPs and effects like that are basically changes in the sequence, either SNPs or deletions. But it's clear now from multiple studies that also these perturbations caused by the Cas9 and the guide RNAs could actually result in structural variation, let's call it this way. So basically, mutational events that are not changes in sequence, but rather changes in the order of fragments or inversions or other types of. So I'm not sure if you're asking this question with the idea of off targets in mind or not, but many people do wonder about whether these structural variants can be problematic or not. And I would simply answer by saying that we know a little right now about structural variation in plants, even independently of the CRISPR technology. The methodology to sequence genomes at the level where you understand their large-scale kind of organization and architecture is still quite expensive, and it cannot be done on, let's say, dozens of mutants or progenies and so on. It's rarely been done that way. So we still don't understand even how, in the natural conditions, how much structural variation is taking place already, independently of the CRISPR system. So it's not surprising, perhaps, that we do see structural variants, and we really need to know more about that topic.
Yeah, okay, thank you. Another thing is that you rightly mentioned that one of the bottlenecks of applying this technology to a lot of crops, not only the major crops. Yeah. So it's to have a very good transformation system. So that is one of the bottlenecks. So what transformation system do you use? And you have also showed an example of the GRF induced improved transformation efficiency recently published. I have also gone through that paper recently. So what transformation system do you use for your wheat transformation?
So we are not necessarily a biotechnology institute. So we are really more of a fundamental basic research institute. So we haven't been investing much ourselves in improving, let's say, transformation technologies, except when we need it as a tool. Yeah. But I know that many of our colleagues, and also next door at the John Innes Center, they were involved in that GRF paper, and they really, in their hands also, it's really improving transformation or generation frequency, as a consequence, transformation frequency quite dramatically. So I think that's really one way forward. Whether that's going to open up transformation to recalcitrant cultivars or recalcitrant crops, I don't know yet. I think we don't know yet, but that's certainly very promising. In our hands, we are also not very good at the particle bombardment type of transformation, which I know many use, including various collaborators of Tofazzal and also people in industry tend to use that. So we tend to use tDNA transfer, agrobacterium-based tDNA transformation, but that's just us. I mean, we are, again, not a biotech institute. Yeah. But I think my advice is really, if I were to sort of bring this technology somewhere, I would start with transformation, because if you don't have the transformation facility, if you don't have the capacity to really bring this system into the plant, then you really cannot do much, right? So I feel all the other steps can be easily addressed even by collaboration, for example, right? So even the cloning and so on can be done in a lab elsewhere, right? So it's fairly easy to do that. And the downstream, the genotyping and the phenotyping, that's also pretty straightforward. So really the weak link is transformation. So I would really urge more investments in that area of biology. of plant biology.
Yeah. Oh, yeah. It was. So I think that's the most. I think the take-home message from this presentation, maybe one of the take-home messages is that we should have a very, and we should invest more our time and effort on developing a good transformation system for all of the crops that we want to modify with these tools. And then coming to your last part of your presentation. Yeah, it was wonderful to learn the story of the wheat blast that started from the BMC biology paper that you published, both of you together. And then it took to you to develop a collaborative project, and that has been funded. And you have developed a lot of lines with this CRISPR. But I know, because I work on rice, and that's very easy compared to wheat. Rice is diploid, and it does not have that three-genome ABD. So I was wondering, so yes, you mentioned that you applied amplicon sequencing for your genotyping, but through amplicon sequencing, so how do you distinguish that this copy of this genome has been edited and the other copy has not been edited? Because that's just I want to know, because this is my curiosity.
Yeah. Yeah, maybe we were naive and ambitious with tackling wheat. I mean, wheat is really something else. I mean, you mentioned rice, but we use barley, and barley is basically easy compared to wheat. It's the same situation. It's the diploid, and it's not complicated to really do the genotyping. So the answer to your question is, first of all, it's a case by case. So it depends on the gene you're targeting. So if you're targeting a single gene, first of all, there are genes that are not present even in 3 copies. So that makes life even easier. But if you have a gene that's present as a single gene, single copy in the 3 genomes, typically there are SNPs, there are polymorphisms that allow you to distinguish between the 3 homologs, as they call them, right? Then by sequencing, if you design your amplicons and your primers in the right way, then you will cover at least some sequences that are that allows you to at least start deducing perhaps which ones have been mutated and have been deleted. It's not straightforward, though. It's really not straightforward. So but I mean, if you want to be a little bit, maybe I'm going to answer your question from different angles. So I mean, the simple answer to your question is it's not easy. And it's not always possible. When you have a complicated gene family like the one we have, we really cannot easily say which copy exactly we have, mutated. If you look at that, there's 7, 5, and 4. So actually you have 16 copies. And I cannot tell you with confidence which ones are actually mutated in which line. But to maybe step back and answer your question differently, perhaps the genotyping is not so critical. Maybe one should go straight to phenotyping. And when you have a positive phenotype, becomes almost like a combination between CRISPR and classical breeding. When you have a favorable phenotype, then you can go back and bother, and maybe in that case, you even sequence the whole genome of that line, So I think, and this is actually the approach we decided to take, because we realized that genotyping every one of those 6,000 lines is just not realistic. And it's not actually very useful, because you don't also necessarily know which mutation is going to be beneficial. In some cases, but in this case, we have very little knowledge about the system. This is really a hypothesis that the rice genes will have the same phenotypes in wheat, but that's a hypothesis. So we don't know precisely which mutation will do what. So in that case, we decided to alter the pipeline and go straight from the CRISPR transformation mutagenesis to phenotyping, and then take the positive lines and take them to further generations, hopefully find favorable phenotypes, and then focus on those lines to identify what mutations they carry. So that's an kind of an alteration of the classical pipeline. If you look at what we did with that tomato, then it's very easy, because you have a single gene, it's tomato, there are only 2 copies. We want to make a knockout. That was not complicated to pick up the mutant and then phenotype it. But maybe in other cases, we can just straight go to phenotyping and worry about the genotyping afterwards.
Yeah. Yeah, I loved your this suggestion of this approach. First, you go to phenotype, and if you get a good and desired phenotype, then you analyze the genotype. That what genotype actually gives that phenotype. So I think
Yeah,
that was the that.
I can
Yeah,
tell you actually a little bit an anecdote there because my colleague Jonathan Jones next door, so he targeted with at the very early days, he targeted this gene, maybe famous one you heard of called DELA. I
yeah,
don't know
yeah,
if some of you know about DELA. So this is involved in gibberellin.
yeah,
And DELA is called DELA because it has this amino acid motif DELLA, right?
yeah.
And so he targeted DELA in tomato with a single cut that is targeting actually that particular motif, so that region of DELLA. And when they did that transformation, one of the tomatoes, this is the Norman Borlaug gene, right? This is the green revolution gene that gives dwarf plants. So one of the mutants they found, they were, when they transformed, they were looking at their tomatoes, and one plant was dwarf. So that's just like so they obviously picked up that plant, sequenced it, and turned out that plant had a 3 base pair deletion in the DELA motif. So it became actually DELA, not DELLA. And so that was basically normally knockout in that gene is actually not viable. So so this was so sometimes phenotyping first. So in that case, that was phenotyping first, right? And sequencing after. So phenotyping first can be valuable. Sorry for adding that anecdote.
No.
It illustrates the point, I think.
Yeah, I understand. So, yeah, it was a treat to listen to you, Professor Sophien Kamoun. So, on behalf of BD Science Porter, I thank you for your nice presentation. Wonderful, and hope to listen to you again soon. And now coming to Professor Islam's presentation. So, Islam's presentation was more about taking through the history of food security and how we are threatened in this climate change scenario to feed our ever-expanding population. In order to keep pace with this population, we need a new breeding technology. And Professor Islam rightly pointed out that CRISPR-Cas9 is one of that new breeding technology that could help us to develop climate-smart crops to resist abiotic stress, to develop pathogen-resistant plants, and also to nutritionally modify plants, and also to, in order to feed the billions, also to domesticate orphan crops or wild relatives of the crops. So it was nice presentation from Professor Islam's, and then he focused on many. His presentation was like a packed presentation, of many aspects. So for you, I have a few questions, and then it was nice to listen to that. So another aspect important to detect a pathogen in its early infection stage. So you developed a collaboration with, I think, Iowa State University and Chinese scientists to develop a detection kit for wheat blast, and that was a wonderful development, obviously. And so my question regarding that is, so what is the status to mass produce that strip, detection strip, for being used by researcher or maybe farmers to detect that pathogen in their field? So what are the status in Bangladesh now?
Excellent. Yes. Thank you very much, Dr. Mullah, for a nice question. Yes, it is a very interesting progress in collaboration with Ohio State University, Chinese Academy of Science, and USDA. And what we are now negotiating with a company to make this, strip available for the practical use. So already 2 companies applied to our institute, and next week we shall sit together with our Vice Chancellor to make a memorandum of understanding with a company. Out of 2, we shall select one. One company is actually Bangladesh USA joint company, and they have a practical experience of developing similar strip, not exactly Cas-based, but other strip, streptavidin, biotin, conjugated strip. These strips are available in the market. The most important point is we need to use large-scale production and validate them in the non-technical hand, the field level as well as the quarantine office, and it must be cheaper so that people can afford it. So we are now in that way, and our university is very encouraging. And if a fund is required, our university will also co-fund with the company. So we are now in line with that, and maybe if we can make it practically available, which is our dream, that would be very good. Thank you so much.
Oh, thank you, Professor Islam. So I wish your dream may be successful very soon. And it would be a very excellent contribution to the community to detect the pathogen. So another thing I would like to thank you for your nice concluding remarks. So you said the bottleneck of translating this technology to the field lies with the regulatory bodies. If they allow it to be cultivated like non-regulated one and as not treated as GMOs, like USDA is doing, as you have cited example of a lot of crops that have been deregulated by USDA. So, and also you mentioned that the theoretical aspect of this should be included into the curricula of school students, college students, and obviously the higher studies. So as a director of your institute, what are the steps that you are taking with the government, Bangladesh government, as you are in many core committees? So what are the steps you are taking to include these CRISPR-Cas technology to include in the curricula of those school students? This is my one question. Another question, just let us know, we developed, this is for Professor Sophien Kamoun, we developed BT brinjal in India, and we could not commercialize that. And now Bangladesh did it. That was an excellent effort from Bangladesh. And I heard from a lot of international scientists, they always cite example of Bangladesh, that how Bangladesh is progressing in science and technology with an open mind to accepting new techniques. So I'm just wondering, this is my second point. I want you to highlight just what Bangladesh is thinking about these gene-edited crops. What would be the future expected regulatory status?
Excellent. Two questions are very interesting. Dr. Kutub, you know that he received a fantastic chapter from US, from Eric. He submitted a curricula package of gene editing for high school student, and very nice in vitro gene editing, showing at the school level, high school level, or like that. And this is really interesting. And we had recently a Zoom meeting. In COVID-19, Sophien initially mentioned that COVID-19 is a big threat for us, for our survival, but offering us some opportunity also. For example, this virtual meeting. So we had a very nice meeting with our agriculture minister and all, education minister on updating the curricula of our, universities, especially in the field of agriculture. And I proposed two things: one, nanotechnology; another, gene editing technology. And all vice chancellors from a different agriculture university, they were attended, and they were convinced. That means, Bangladesh, in one sense, Bangladesh is a developing country. We have lots of problems. But in other sense, if you consider the acceptance of advanced science, Bangladesh is smart enough, as you mentioned, in case of BT brinjal, developed by India, tested in the field, and we are now in the practical life. 65,000 farmers cultivated last year in our country, and 17% of our total brinjal are now BT brinjal, and farmers are getting benefit, no pesticide spray to control the brinjal shoot and fruit borer. Our agriculture minister, that he was a chief scientist of our Bangladesh Agriculture Research Council. And he is a very big fan of biotechnology. This is why it is a good point. Our last agriculture minister, Motia Chaudhuri, she always told us we eat science. So science is the only tool to address our challenges. We have limited land. We have lots of population. So no way other than accepting science. So where we need science, whether it is gene editing or other technology, no problem. In the National Policy Committee, I raised twice to make a decision on gene-edited crop. When I explained about the gene-edited crop, no transgene there, and at that time, all the members asked me why you need a regulation. If no transgene, go to the field. I don't know exactly what would be the future. What is a good point is recently our government and Canadian government had signed a big memorandum of understanding to collaborate with Global Institute for Food Security. And I am the team leader of genomics and phenomics, where I included gene editing. But government said if gene edited product is free from transgene, then we can go straight forward, forgetting what people are doing in other countries. So there is, I think, high promise in Bangladesh. If we can really solve a real life problem like wheat blast, obviously it is a terrible problem, as Sophien explained. Wheat, transformation and engineering of wheat is not easy. A few months ago, Ulf published a paper. Wheat is abandoned in genetic engineering, actually, abandoned crop. It was published in Science. So in case of wheat, if we can make a breakthrough, we are trying our level best in many ways, then it would be a revolution in Bangladesh. Revolution not only in terms of economic impact, but the application of technology in a complicated subject, frontier technology. So I am, a super ambitious man. So I always try to inspire the young people who will make the real impact. This is why in my university, I included CRISPR-Cas technology in undergrad level curriculum. So our academic council approved it. I introduced a course at master's and PhD level. So trying our level best. I hope other university will follow it.
Great to listen those development. So just before I just transfer to Dr. Burhanuddin, just I want to say, so I heard from Professor Sophien that it was a slip of tongue, he said instead of CRISPR, Christmas. So it's really CRISPR technology is Christmas for all the biologists. It's all over the year. It is a Christmas for biologists. So thank you, both of you. And it was nice to interact with both of you and listen to you. Now the mic is to Dr. Burhanuddin.
Thank you, Dr. Kutubuddin. Can you hear me?
Yes.
You're audible, sir. Yeah.
Okay. So before going into any technological details, I would like to ask a very personal opinion from Dr. Sophien, like what is he thinks about the safety of genome-edited foods? Like what should be the regulations? Like should be always DNA-free genome editing, like with the RNA and ribonucleoproteins, or it can be with DNA simple, like usual cloning vector-mediated genome editing? What is your personal opinion about food safety, biological safety of the foods?
I think it's I mean, I can give my opinion, but obviously different countries have different approach to this. I mean, and personally, I think the scientific approach to this question is to regulate the product, not the method, right? So what really matters is the plant you're putting in the field. What is it? Well, how is it modified? If it's a few base changes or a deletion or even a big deletion, that might be good, right? I mean, all these anti-GM people, all these people against transgenic technology, they've been telling us all these years that putting genes in the plant can be dangerous. So what if we remove 4 genes, like in our deletion there, that 50 kb deletion? Isn't that making the plant safer? You could argue that there's less chance that people will have allergic reaction to those 4 genes removed, right? So I think more seriously, I think the key point, I think, is to regulate the plant and not the method. It really doesn't matter. I mean, how you reach the mutation, how you make it through breeding or through mutagenesis or through CRISPR. If that plant is safe, then it should be grown, right? So sorry, say again.
Yeah, that's for sure. That's for sure. Like you want to see what's the end results.
Yeah.
But the thing is that with the normal transformation stuffs, there's a possibility that the foreign DNA can be incorporated irreversibly to your genome of the plants. So how will you address this concern?
No, I mean, this is exactly what we did with that Tomelo study, and I can share the paper. In that case, that was through tDNA transformation, but because the tDNA was inserted in a different region of the genome than the MLO mutation, then in the next generation, this segregated out. And so we could quickly generate plants that don't have the tDNA but carry the favorable mutation.
Yeah.
So in plant biology, unlike, let's say, human biology, it's actually fairly straightforward to do these things, to basically segregate out the tDNA from the mutation, the favorable mutation. So in that case, again, if you're regulating the product, then that plant that doesn't have any tDNA left, and we in that paper we actually sequenced the genome of that plant, of the mutant, and we showed that there was no agrobacterium DNA left in that particular plant. So this can be done. I mean, this is again different methods, different approaches. But again, I think if I'm a regulator and I'm a logical, rational, scientific-minded regulator, I would regulate the product. I would not regulate the method.
Okay, thank you.
So I think I encourage regulators to think that way. I don't know if there are any in the audience, but
yeah. So okay. So now, I mean, the thing is that I have worked with the human cell lines. It's really pretty straightforward. You have 2 copies of the genes, and even if it's cancer cells, it will have probably 3 copies. So my question is that in one of your slides, you have showed that there are multiple copies of genes in wheat. So have you tried any high throughput dot blot techniques to see whether any proteins is just removed, like protein injection methods, high throughut techniques? Have you tried, or is there any good antibody for that?
Yeah, no, I mean, unfortunately, this is not like human systems, so we cannot just go and buy antibodies and this sort of things. But this is something we haven't done yet because we wanted to first identify the favorable lines and then analyze them in more detail. So at this stage, we haven't done that in this particular project. Okay. But maybe the irony here is, again, I'm going to answer your question a little differently. So the reason why wheat actually is a hexaploid is because those 3 genomes were actually brought together by humans by breeding and by domestication of wheat. So wheat in itself is actually a very strange plant that does not exist in the wild. But those 3 genomes were actually hybridized by humans throughout domestication of wheat. So it is a wonderful plant, obviously, produces a lot of calories, has been critical to actually the fact that we have 7 billion people on this planet. But it's actually a freak of nature that's man-made. So that's an interesting aspect of it.
I mean, so I have really very few experience with plant stuffs. The only thing I can remember during my study in master study in Wageningen, where I did like inducible hairpin to knock down genes. I mean, what is your personal opinion in the cases where you have multiple copies of genes? Isn't it straightforward to have a hairpin which you can induce and then the RNA level goes down and protein level goes down, just only for the multiple copy problems?
No,
What do you think?
totally. I mean, so I think there's two answers there. I mean, first of all, the multiple copy just slows things down, but it's still possible to knock out multiple genes. It's, in fact, I think in many of the mutants we have, we think we deleted the whole cluster. So it's actually possible to just remove the whole thing, right? So it's not necessarily a big problem. But the hairpin and gene silencing, as you know, RNAi was actually first discovered in plants. Sorry to our Nobel Prize that forgot about plant biology, but anyways, that's fine. So hairpins and gene silencing in general are used a lot in plant biology. A problem with commercializing, I think, crops with hairpin and with silencing is, first of all, they are viewed as transgenic, as GM. So there is that resistance against using transgenics in agriculture. And second of all, sometimes there are problems with stability. So they are sometimes, they do revert back, or the effect reduces over time or something like that. And so that's the second issue. So, but there are I mean, this is another approach to knock down genes. And as you mentioned, exactly precisely why we have many projects where we like to use gene silencing or hairpin or virus induced and so on. I think there was actually a question in the chat. We don't have time to answer all the questions. There was a question about how it compares to gene silencing. Well, this is exactly one advantage of gene silencing. Is you could target multiple genes at one go.
Okay, so thanks, Dr. Sophien. I'd not hold you for long. I think people also want to ask a lot of questions. I have one last question for Dr. Islam. Dr. Sophien? Okay.
Yes.
Okay. So, I mean, since I have seen that you have developed this kit for detecting fungus, so you know the Dr. Feng Zhang's lab had developed the Sherlocks. It's like a very straightforward technique. Have you anything in your mind that you want to try, at least in our country, to detect the coronavirus with these Sherlock techniques?
Coronavirus dangerous. You know, I am confined to my house. I'm really afraid of it because the reaction of coronavirus in different person is different. We don't know its biology. Two of my friends, they were killed by coronavirus. And in our country, you know, to handle coronavirus, we require a laboratory biosafety level 4 or like that. So in our institute, we are not yet thinking to handle the coronavirus at all, because we are mainly focusing plant and microorganism. But Sherlock technique already been established. We can introduce it into Bangladesh, because some companies already scaled up it. And if our government approve it, then it could be used in Bangladesh. Lots of other, diagnostic tools, rapid diagnostic tools have been developed for coronavirus.
Yeah, I am completely agree with you that it's really hard to handle with the virus. But I mean, in the paper that they say that if you just put some virus neutralizing media and then put it outside in the lab, then you can really use it for detection.
It can be used in our, established coronavirus testing laboratories, because we have now more than 100 coronavirus testing laboratory nationwide. So if government approve it, then it would be no problem to use it, because it is now commercially available. I followed that, discovery.
I mean, yeah, of course, because since you have, you can pursue people. I mean, it has the rapid nature of the protein-based kits, but it is nuclear amplification kits. So I think it's really amazing if we can implement it here in our country. Of course, thank you for the fungal detection kit that you developed. Congratulations again. And I mean, of course, I think. I think it's really time for us now to think about, even if we cannot do the basic molecular stuffs in our country, but at least we can invest so that we can employ or you can exploit some techniques that other people developed in our country by investing some money. Thanks everyone for nice talk. Thanks Dr. Islam for nice presentation, and thanks Sophien and Kamoun for really very enlightening sessions. Thanks everyone for inviting me as well.
I want to add one point. Dr. Sophien and Mullah. Dr. Mullah both are experienced enough in application of CRISPR-Cas technique in plants. In Bangladesh, what I am doing now, I am pursuing our government to train young people and getting good funds as well as good people to train our young people. So in that case, I may ask Dr. Mullah and Sophien to help us because government seems to me very positive. As in that case, if we can train a few hundreds of young talented people on using CRISPR-Cas genome editing technique in plants or other organisms, then you can see young people, they have lots of innovative ideas that can make a robust impact of this technology in our country. So hopefully we shall have some good news from our policymakers. I am now working to shape this one. Recently, during coronavirus, we had a coordinated meeting on National Biotechnology Group, and they advised me to design a curricula for such a training and seek for funding. Let us see what happens in future. I am very optimistic, and I hope Sophien and Mullah both can support me.
Thank you, Tofazzal. And I mean, this is actually a good transition for me to do a little bit of advertisement, which I think is appropriate for our audience. The Sensby Lab, my institution, will be starting a new master's course next year in global plant health. And so this should start in 2021, hopefully, unless we get delayed with this coronavirus stuff. But the good news for everyone is we have generous fellowship scholarships from David Sainsbury, who is the benefactor of our laboratory, who will support international studentships for fellowships to attend this master's course. So stay tuned. We will advertise this in due time, but I thought I would let you know about this. I'm personally, and I know that my colleague Nick Talbot are very excited about launching this new master's program in global plant health.
Thank you, Dr. Kiribin Lamella. Thank you, Dr. Burhanuddin. Thank you, Dr. Sophien Kamoun and Dr. Tofazzal Islam. And the biggest takeaway for this seminar would be if we can train a lot more students from our country to use CRISPR-Cas9 for talent and other advanced techniques to develop more and more disease-resistant species and different crops varieties as well. So now I'd like to request our event chair, Dr. Abdullah Mohammed, to share a few words about this new platform that he has started. And also I'd like to ask him to give us a remark about the seminar that we're doing today. So Dr. Shual, sir.
Bismillahirrahmanirrahim. It gives me great pleasure to welcome all of you to join our Shaping the Future virtual seminar series. Assalamu alaikum, good evening, good morning, and good afternoon, wherever you are. It's so wonderful to see you here. On behalf of Science Portal Bangladesh, Cell Genetics and Plant Biotechnology Lab, and as the chair of this seminar, I would like to thank our outstanding speakers, our guests, delegates, journalists, and students who have joined to grace this seminar. First of all, I would like to extend a special welcome to our guest of honor, Dr. Sophien Kamoun, who is a senior scientist at Science Bureau Laboratory and professor of biology at the University of East Anglia. He is known for his seminal contribution to our understanding of plant disease and plant immunity, having pioneered genomics and molecular biology methods to reveal fundamental insights into the biology and evaluation of eukaryotic plant pathogen. And we heard from him excellent and very excellent talks about the recent advancement. With us, a famous biotechnologist, Professor Dr. Tofazzal Islam sir. He is a director of the Institute of Biotechnology and Genetic Engineering. Those who know him, you know that he is very dynamic and very kind-hearted person who always try to collaborate and motivate us for doing better things. He is now Professor of Bangabandhu Sheikh Mujibur Rahman Agricultural University, Gazipur. All of we from Bangladesh know him very well. Our distinguished panelist, Dr. Kutubuddin Ali Molla, he is senior scientist, National Rice Research Institute, India. He is a very dynamic, well-known scientist in genome editing as well. We have with us Dr. Borhanuddin, Associate Professor of Biochemistry and Molecular Biology, Jahangirnagar University. We are so excited to have you all here to participate in our virtual seminar, CRISPR-Cas9, New Frontiers Technology for Future Agriculture. This event is jointly organized by Science Portal Bangladesh, Cell Genetics and Plant Biotechnology Lab, Jahangirnagar University. We already knew and we already learned that genome editing, also called gene editing, is a group of technologies that give scientists the ability to change an organism's DNA. It's very simple to elaborate, but a little bit tricky to work. These technologies allow genetic materials to be added, removed, or altered at particular locations in the genome. Several approaches to genome editing have been developed. A recent one is known as CRISPR-Cas9. It's not a recent one. It's already have several years. It's now, I would like to say, it's now young age right now. Which is short for clustered regulatory interspaced short palindromic repeats and CRISPR-associated protein 9. The CRISPR-Cas9 system has generated a lot of excitement in the scientific community because it is faster, cheaper, more accurate, and more efficient than other existing genome editing methods. That already explained Professor Islam and Dr. Kamoun. And we knew that the achievement of CRISPR-Cas9, what has happened is Nobel Prize already got these technologies. CRISPR-Cas9 was adopted from a naturally occurring genome editing system in bacteria. The CRISPR-Cas9 system works similarly in the lab as while this is happening. Researchers create a small piece of RNA with a short guide sequence that binds to a specific target sequence of DNA in genome. Genome editing is of great interest in the improvement of crops and other plants, prevention and treatment of human disease. Currently, most research on genome editing is done to diagnose and understand diseases using cell and animal models, beside the plant models. Scientists are still working to determine whether this approach is safe and effective for use in humans and the environment. Science Portal Bangladesh is trying to reach general people with the solution by cutting-edge technologies. I would rather say cutting-edge toolkits that Professor Tofazzal Islam already teaches us in a simple and easy to understand way. To fulfill one of our challenges is to develop genome editing as a frugal science and also citizen science. We really appreciate your kind help and extended hand to Science Portal Bangladesh from Professor Islam, Professor Sophien, and Dr. Molla to reach our goal. We really want to reach general people in their own language to explain them what is CRISPR and how this revolutionary technology will help us in future for food or feed in other way. I would like to thank our members, sponsors, well-wishers, and all those who have given their active support to organize this seminar. This seminar will add extra values to our ongoing activities during the COVID-19 pandemic, when most academic activities are suspended. I hope that today inspires ideas and discussion around the ways that we can secure the food for the lives now and the future. Thank you again to everyone for being supportive of the Science Portal Bangladesh platform, and I hope that you will follow us to have more exciting and enriching academic sessions in the coming days. Thank you. Now, I would like to start this most exciting QA session. I have several questions with my hand, and the rest of the question, Sam will help to address. First question to Professor Islam, sir. Sir, are you?
Yes, I am hearing.
Sir, the question is how CRISPR should be used. Actually, the questionnaire should be used in Bangladesh perspective. That means I think he or she may be asked in which way we can safely use this technique. So, would you please address this issue?
Yes, CRISPR is, its application is versatile. It is a toolkit, can be used bioediting of any organism. But obviously, we should not start it editing of human germline or like that, because it is not our priority area. Our priority area is, to ensure the food and nutritional security. This is really a big challenging issue for Bangladesh, and our Prime Minister, she told we should not keep inches of land uncultivated or like that, because land is very scarce. In that case, CRISPR, I think, should get top priority to use in agriculture for developing climate smart crop variety. But it should not be limited there only. But we can do lots of basic research in microbiology and other using the cell line or others. So CRISPR, I am dreaming to see CRISPR in every biology lab, and young people will use it. And it is an emerging technology, not yet matured enough. Always it is changing. So CRISPR, I want to see it everywhere. We need to introduce CRISPR in our curricula at even secondary school level, university, undergrad, all biology level. And in that case, like Professor Suhail and other, people who are leading biotechnology and biology, you have to, make it a popular topic and convince the policymaker so that we can achieve this dream. And obviously, I, as a fan of this technology, I believe it has a big power to make a change in our society, which we are expecting.
Yes, sir. Science Portal Bangladesh is really is very much keen to work on that, because we'd like to reach general people. I mean, not only academicians, but general peoples like farmers, like day laborers, and end users about this technology. So we would like to go to them, and we will design our own way to convince them about this technology. And we would like to engage with this technology, some innovations and implications that will definitely help us in future to adopt those technology very easily, crossing the all hurdles, I believe, like awareness program or some sorts of teaching learning programs, or some sorts of one-to-one take-home message, or such types of program. And in that case, definitely we will seek your kind help, as well as Professor
Professor Suhail, I want to add one point. In 2016, when I faced a big, crowd of our agriculturist policymakers, they asked me, how can we solve the problem of wheat blast? Because you, figure out what is it that is the origin of the pathogen, but how can we solve it? Then Sophien, actually, I had a very good discussion with Sophien. Sophien told me CRISPR could be a tool. And at that time, I didn't know much about the CRISPR. And I explained about the CRISPR. And there were, lots of international people from international bodies like USDA, FAO, and many others. And all were pinned off silent. Didn't ask me any question how it is possible or like that. And finally, our chairman of Bangladesh Agriculture Research Council, she told us that it seems like CRISPR a blue sky for Bangladesh. But at that time they didn't offer me funding. Sophien and Nick, they managed funding from the BBSRC. But just after having that fund, Bangladesh Agriculture Research Council invited me to submit a project on CRISPR to offer me the funding. That means when you, discuss in a way your idea so that they can and convince them, then it would be nice. So after 2016, I traveled almost all big universities in Bangladesh, including your Jahangirnagar University, to talk about the CRISPR. What is it? But now I believe it is a nice ground to work and to start work in our every laboratory. Even in vitro, we can do the gene editing. I shall share with you some, protocol. Maybe in vitro, in the practical class, you can train undergrad student to do, bacterial gene editing or other in vitro DNA cut or, mutagenesis and other things. So let us work together and hopefully we can make a big change. It is obviously new because all the, senior people, they didn't know it in their curriculum. It was not included as it is an emerging technology. Young people, especially who are attending in this program and especially, science portal, you can make a big change.
Thank you, sir.
Thank you so much.
Thank you, sir. Next question to Professor Kamoun, sir. What are the areas beyond susceptible gene editing for developing disease-tolerant variety?
Yes, so I had the slide on that, but I decided not to spend too much time discussing the other approaches. But the main approach is really maybe a combination of gene editing and transgenics, which is to bring in new genes in a specific locus. So this is known as gene stacking. So if you have, for example, 3 or 4 resistance genes that are known and cloned against your pathogen, for example, we have many resistance genes cloned in rice against rice blast, and some of these may function against wheat blast. Then you could introduce them all in a single locus using the CRISPR technology. So it's not truly editing per se, it's actually using editing or CRISPR technology to really introduce genes in specific locus. And there can be many advantages of having all these genes together in one locus. So there are technical advantages and also easier afterwards to breed them into other varieties. But there's also the introduction of point mutations in resistance genes, in immune receptors, for example, that can improve their function. This can be done. Also mutations in promoters, which could enhance the expression of a particular gene that is beneficial for the defense response or for the immune response. So there are a lot of little tricks like that. But they generally, again, require more knowledge about the pathogen system you're studying. So the advantage of, I would say, if I had a dream, if I had a magic wand now, scientists are a little bit like magicians, right? So I know we have a broad audience here. So science is actually the real magic, right? Magicians, they are lying, right? They are confusing you with the tricks. But actually in science, we do real magic. So the CRISPR is a little bit like magic, actually. you are introducing mutations specifically in that place in the genome. But if I had the magic wand and I had a wish to make, I would wish to have a gene like MLO for every kind of pathogen. If you had an MLO for blast, if you had an MLO for Phytophthora, if you had an MLO for rust fungi, that would be great, because that mutation is a loss of function mutation. You delete the gene. Generally, there is no other phenotypes in the plant, and then that mutant is resistant to powdery mildews, that whole class of pathogens. So, and I would say, as a plant pathologist, I predict these genes are there. We just don't know which ones, right? So if we had an MLO-like gene for every type of pathogen, I think we would have quite an amazing advances in our field. Incidentally, there was a little bit an anecdote I forgot to mention, but I told you about that urban garden, urban tomato. So it turned out that that's very susceptible to powdery mildew. So last year, a colleague, Professor Xian in Maryland, he grew it and he was testing it as part of an outreach program, and then it got completely colonized with powdery mildew. So next year they will be introducing a fourth mutation, which would be the MLO mutation in that urban tomato. So that's also, it shows the power of the technology. You can keep adding traits as you require them. Thank you for the question.
It wondered me about this is urban tomato. So how about rice or wheat? Can we make such types of changes? Then we can make urban rice, we can grow in the rooftop, or we can get the bunch of rice or wheat. Is it possible, sir?
You know, I think this is exactly the kind of out-of-the-box creative thinking that we need. And I don't know, but I'm sure in our audience today we have some young people who can start dreaming about this kind of ideas. So maybe it is possible. I think you probably have a better understanding of the urban situation and maybe the space available and all that, and what would be the ideal crops to grow there. I mean, even dwarf trees or this kind of things. You know, I don't know what is most appropriate, but this is exactly, I think, what I encourage. You know, I'm a strong believer, Tofazzal knows that, of the bottom-up approach. You know, if you bring a group of people together who are dynamic and enthusiastic, then great ideas will come out of that. So I think that's what we are doing today, and this is what's so exciting. So I really encourage the young people, especially here, to think and dream about ideas like that. And who knows?
Yes.
If you don't dream, it's not going to happen, right?
Yes, yes.
Right.
Sir, next question for you as well. How can we adapt the CRISPR-Cas9 system in plant nematode interaction research?
I mean, that's a very specific question. But of course, in plant nematology also, you can imagine there are susceptibility genes also, that the nematodes are known to have effectors that are used to cause disease and colonize the plant. And so they have targets. And I would imagine that's the same concepts I talked about can be applied to nematodes. Again, we need more fundamental research there to understand these systems. But in principle, everything I mentioned can be applied to any plant pathogen or plant-based system.
Thank you, sir. Next question to Dr. Kutubuddin. If you are, please unmute yourself. Scope of using advanced gateway cloning using CRISPR.
Advanced gateway cloning. I think Professor Kamoun would be the perfect person to answer, because
please, sir.
Yeah, he was using. He mentioned about gateway cloning.
No, I would say not gateway.
Gateway cloning.
Golden Gate.
Oh, Golden Gate. Sorry.
Yeah.
Sorry. Okay.
But that's fine. I mean,
Yeah.
Yeah. Someone asked.
I can put a link
sir.
to the publications in the chat. So for
okay, sir. Okay. Thank you. So our next question. Again, with rice, maybe Kutubuddin, you can answer this one. What is the status for the development of disease resistance rice variety by using CRISPR-Cas9 technology?
So if I want to mention one very distinct example that also goes with the line that Professor Kamoun discussed in his presentation, that is susceptibility gene editing. So rice has also known susceptibility gene for bacterial blight pathogen. That's called SWIT group of genes, And recently, there was publication in 2012 with talent-mediated inactivation of SWIT genes. It's not inactivation, but it's promoter editing. Because bacteria secrete a protein that's called tail protein, and that binds with the promoter of that SWIT gene to express highly that SWIT gene. And the product of that expression is actually being used by the pathogen to better colonize the rice plant. Okay, so if we can just interrupt that interaction of tail protein with the promoter sequence of SWIT genes of rice, then we could grow, we could develop a resistant plant. So that has been also utilized, that approach by CRISPR-Cas recently in Nature Biotech last year in 2019. Ricardo Oliva, and it is an international collaboration. They published a paper and developed broad spectrum bacterial blight resistant rice varieties using CRISPR-Cas9. So they targeted SWIT11, SWIT13, and SWIT14. And all promoters have been edited, and they generated that line, and that showed excellent level of broad spectrum resistance against African isolates, against Asian isolates. So that's a great example of using the same approach as Professor Sophien has discussed. Yeah. So I can also share that publication link in the chat box. Yeah. It's already Ricardo Oliva paper have been posted by Professor Sophien Kamoun. Thank you.
Thank you.
And a little bit of pride there, because Ricardo Oliva was a postdoc in my lab before he moved to Erie. So I'm very proud of him and his success.
Okay.
He's my son.
Next question to Professor Islam, sir.
Yes.
This is practical question for Bangladeshi students. What kind of resources, computational and wet lab facilities are required to implement the CRISPR-Cas9 for the crop development, particularly disease resistance or other crop development program?
Facilities for CRISPR-Cas actually not too much is required, but I guess you have most important is your target should be specific and basic knowledge. If you have, then a modest biotechnology lab is okay. As Dr. Mullah mentioned, transformation protocol. This is also critical. So in case of transformation of your target crop, the protocol needs to be established, and tissue culture or other ways that you must have such facilities. PCR obviously you have, and I think not too much facilities is required for starting the job.
Okay.
You need to have, for example, disease resistance. So in that case, you need to have, culture facilities for the pathogen, as well as the incubator for doing the disease resistance assay. In that case, Sufyan's idea is very smart. You do not need to have everything in a single lab. The smart idea in doing any biotechnological job and facing any challenge, to collaborate with several labs. For example, in our case, we collaborate with plant pathology lab, because plant pathologists, they know how to handle the microorganism and do the pathological work, test the transformants or like that. So if you want to do all in a lab, even in case of disease resistance, you need lots of things. But if you have an interdisciplinary, collaborative job with several labs, so several experts can complement their experiences and expertise, as well as share the facilities. For example, in determination of wheat, genetic identity of wheat blast fungus. Sophien, you can remember how many people, some people in the field collecting the sample or like that. So you have seen 31 people engaged in a paper, and within weeks, we showed the result, and it was super fast. And within 3 or 4 months, you have seen the published paper. No funding, no project. So idea is most important, I think. Rather than arranging all the big equipments in a lab at first, and then you think to start something. Start something with whatever you have now, and communicate with other groups who have other facilities to complement with you. Then we
okay, sir. Let's make some business, sir. Like in my lab, we have well-established tissue culture facilities. And we have a very nice rice tissue culture regeneration and acclimatization systems we developed, and all sorts of tissue culture facilities we have, monocot and dicot systems both, and we can have experts, including myself, transfer. How can I collaborate with you, sir? Because I don't have...
Excellent. You can collaborate with us, because in case of tissue culture, our lab is not, powerful enough. We have tissue culture lab, but what can you do? You can use our facilities. We have Illumina sequencing platform, real-time PCR, gene gun, all other things. So it is easy to make a breeze, because Jahangirnagar University and Bangabandhu Sheikh Mujibur Rahman Agriculture University not so far. So we can divide our job, and if you have a good idea, and if it is nationally a priority topic, funding would be, I'm sure, no problem. Since 2010, I didn't face a moment of scarcity of funding. You can ask Sophien. Sophien told me, please go and collect the samples from the field. I have fund. But I didn't ask him to, deliver fund from UK to Bangladesh, because I didn't face any problem of doing any research with a good idea, and it was not halted due to lack of funding. In last 10 years, I can guarantee you, even in recently, Bangladesh Academy of Science forced me to accept a funding of 2 crores taka, a commission project. I didn't apply for the project. They say, you must do this project because this is nationally important. This is why I formed a big group, interdisciplinary group. They are going to do the job. Their project is very big project, food and nutritional security under COVID-19.
Thank you, sir.
They offered big fund to see. So funding would not be a big problem. Please, I welcome you as well as any other scientist. Please come with a good idea, which is nationally important. We can find a way. Even in my lab, if it is not enough, I can talk with Sophien. I can talk with Guliang Wang. I can talk with Mullah, and we can make a big change.
Thank you, sir. The next question, Dr. Kamoun, how much time it generally takes to complete a successful round of this technique? I mean, from the beginning to get a product, usually how much time it's required, depending on, let's say, like wheat or rice or like tomato. So how much time?
Yeah, it's a good question. And the point about the different crops is a good one, because depending on the plant and the crop, the transformation procedure takes different period of time. But this is obviously something we were concerned about, and we documented very carefully in the study on MLO. So I will put the report, the paper in the chat. But in that particular project, it took us exactly 9 months from the moment we started the cloning to the moment we had actually a line without the tDNA in it. validated. So it can go really, really fast if you have, of course, a plant like tomato that is fairly straightforward, and you have a strategy that is straightforward. So this can go dramatically fast. Let's put it this way. With wheat, it's a little bit slower because of the complications of the transformation takes more time, but also the follow-up work after you have the plants is also more complicated, as you heard, with the complexity of the gene families and all that. So, but it can be, as little as 9 months, really. And again, I encourage the person who asked the question to check the paper. I will put it in the link in the chat. Sorry.
Please follow the chat box, and you will get the details about this. So, Dr. Borhan, are you with us?
Yeah.
Dr. Borhan.
Yes, I hear you.
Another question.
Can you hear me?
Yes. And another question: how CRISPR works in vaccination? As you had experience on animal systems working with CRISPR, so in case of vaccination, will it work, this CRISPR system?
So the question is, how can we use CRISPR techniques for vaccination?
Yes.
Okay, so honestly speaking, I have no idea about that. But I think if we want to have to modify a protein or antibody, I don't know. I have no idea. I don't know whether anybody is trying CRISPR technology for vaccination as well.
Okay. So, Tofazzal Islam sir, CRISPR-Cas line has more importance in agriculture and fisheries sector. Is it true or false? Someone?
Yes, it is obviously true. And
agriculture, we knew that. How about in fisheries sector?
Yes, in case of fisheries, lots of papers are available. Even gene-edited fish are also exist. So obviously, I think it is in agriculture and in ensuring food and nutritional security. We need such a powerful tool, because the tools which we have been using or used for making the green revolution in the past several decades, they are now blunt and not showing. They are not showing high promise as they showed before. But we need to increase our food production very fastly. In Bangladesh targeted to increase food production 100% by the year 2041. So by using classical methods, you cannot do it. So CRISPR would be one of the weapons that we can exploit.
Thank you, sir. Sophien, sir, can this tool make breakthrough in organic culture, and how?
That's a very straightforward question. I mean, I think actually methods of genetically improving plants for disease resistance are automatically favorable to organic agriculture, because they would reduce, obviously, chemical inputs. If you can deliver genetic resistance, then you don't need to deal with fungicides and so on. So the answer is yes.
Thank you, sir. Tofazzal Islam, sir. So this is, I think, one question, but it is representation of all biotech graduate in Bangladesh. How will I be able to do research in the field of genetic engineering like CRISPR-Cas9 system, where I have no opportunity and equipments or labs in my university or in my institute? That is hardly true for us. So would you please answer this question?
Yes, it is a real question, because I visited almost all public and private universities where biotechnology and genetic engineering degrees are offered, and many of the departments or institutes have not enough facilities to do globally competitive research. It's true. But you are receiving, the course and advanced training theoretically. In practically, it's true. Even in in case of our student, all students are not receiving the hands-on training. But if you are desiring to become a researcher, a scientist, and dedicate yourself for science, maybe I think you can find some ways. In Bangladesh, there are some good laboratories in biotechnology and genetic engineering. So if you have a good dream to collaborate with them, because I can tell you, BRAC University, they have no good facilities for offering a training in biotechnology. But what they are doing, they are collaborating with ICDDRB. So all the students at BRAC University, they are doing their thesis work at International Institute of Diarrheal Disease Research, or like that. So they're receiving a good training. So somehow we have to find the ways. I hosted several students from North-South University and East-West University in my lab, because they had not enough facilities at that time. But biotechnology is a growing discipline in Bangladesh, and we need to go a long way to have full-fledged facilities in every laboratories. So collaboration would be the key, and if you have thirst for doing basic research or making breakthrough in any topic, you need to contact with the right person. There are several people who are supporting the young people in Bangladesh, and many of our graduates also moving to other countries to get advanced training, doing masters or PhD, and then coming back home like that. So keep your wishes alive and try your level best. You can find the way. And I welcome if someone wishes to visit my laboratory and wants to do something meaningful. Obviously, our team will welcome.
Sir, may I add something? As you are working hard, curriculums and other ways, improvement of curriculum and other ways. So, sir, would you please try for an umbrella fund to capacity building biotechnology and genetic engineering labs, at least 10 or 15 labs within 2-3 years in Bangladesh, and I would like to be with you in that ambitious project. So our students, what you are saying, this going outside or getting theoretical, actually they are getting disappointed because that was not their goal. They wanted to learn by their own hand in their mother institute. So even though they would like to go away, but if they can avail their chances in their own institute, it would be very good for them. So, sir, I would like to request you for a drive to raising a fund to at least develop some frugal facilities in the biotechnology departments in Bangladesh to ensure our students that they can get some opportunity to do some research or something in their own institute.
Exactly. Actually, we need to discuss, this topic in our forum. For example, 20 labs, that means from 20 universities can jointly support a project to, our planning ministry, to, enhance the capacity of basic, facilities of the laboratories. This is one way. Another way is you can make a small project, less than 50 crore taka from your department and submit through your university to the planning. And it does not require, ECNEC meeting or like that. And there is a high chance if you can convince your vice chancellor to get the fund. For example, recently we got 1.2 million US dollar funding from our core government funding. So it is possible. In that case, the young teachers who are struggling with facilities and other things, they have to try to find the ways. There are several ways to find a solution. And I welcome you. Maybe we can several groups together. We can submit a project to the planning ministry, and then pursue it because it is for the sake of the interest of our country.
Thank you, sir. So during the conference, we are doing some business also. Sorry for that. Next question to Professor Kamoun sir. How can be CRISPR-Cas technology applied in an adult system for their genome editing? Is it possible, sir?
Sorry, I missed that. In which system?
How can CRISPR-Cas technology applied in the adult system for genome editing? Not from the beginning, I mean like adult plant or like adult. So can be used CRISPR-Cas technology for genome edit gene?
Yeah. So one of the maybe most exciting route of implementing this technology in a very easy way in plants is to use virus vectors. So if you can imagine, so those of you who know plant biology would appreciate this. So if you imagine a virus that can infect the meristem of the plant, right? So imagine now delivering the CRISPR system, the Cas9 and the guide RNAs, through that virus directly into the meristem. So this would be an amazing advance because then all you need is to infect your plant with the virus. The virus will infect the meristem of the plant. It will deliver the Cas9 and the guide RNA. They will do the job of doing the editing in the meristem. And then the seeds you will collect from that plant will be edited.
But so sir, generally we know that meristem, virus can't reach to the meristem. So that this meristem mediated plant regeneration system is developing for virus resistant plant. So how can we do this technique, sir?
So first of all, not all viruses cannot colonize the meristem. There are viruses, for example, tobacco rattle virus can colonize the meristem. So there are viruses that can colonize the meristem. This is it's true that the meristem is resistant, if you like, or immune to most viruses, but some of them still can break through, and those are actually the seed-transmitted viruses, right? So if you think about it, a lot of the seed-transmitted viruses are actually viruses that can get into the meristem. So the trick there is to use some of those viruses. I know the tobacco rattle virus is the most promising one to use for this method. But the problem is that these viruses, like tobacco rattle virus, TRV, is not a very good vector, and it's limited in the kind of size of gene you can clone in it. So there's some excitement now into some new variants, not Cas9, but new CRISPR enzymes that are actually smaller. And so those smaller genes may actually work better in these virus vectors. So you see, again, the possibilities are limitless. We're really at the beginning. So, but if you imagine having, again, a virus-delivered meristem editing, this will completely change agriculture, right? Because now you inoculate your plant with this virus, you collect the seeds, and your seeds are actually a collection of targeted mutants, which will be absolutely fantastic, right?
So far, do we have any results on it, or it's still ongoing?
As far as I haven't seen some conclusive results. I think a lot of people are trying, and there are various papers describing various steps, but it's not yet a routine application.
So, Dr. Kutubuddin, do you know about this?
I think, I read some papers. So I would recommend going through the publication of Professor Daniel Boytes from University of Minnesota. So they are trying of using this virus-mediated transformations Initially, they're trying with tobacco plant, but yeah, I see some recent paper. Yeah.
Okay, thank you so much. And then next question. Sir, Tofazzal sir.
Yes.
Except your organization, as all we know that you are doing this CRISPR or genome editing, except your organization, do you know which organizations are working on such genome editing technology and what are their outcome, including yours?
Actually, Dhaka University Professor Jaba Islam Shiraz, she is doing some basic work on, validating the salinity tolerant genes by using, implanta in rice, implanta CRISPR. She told me recently in a meeting, and she is using there. Bangladesh Rice Research Institute, one scientist, Dr. Horendro, he received a training in China, and they started a project. In our case, also, we are at the beginning stage in rice. We started a project to edit our some very high-yielding variety to make them aromatic by changing the BDH2 gene, mutating that gene or like that. So we are also at the beginning. Actually, the coronavirus really affected us to receive some reagents, chemicals, as well as some constructs from other collaborators. This is why a bit slow down, but we are moving on. And in case of wheat blast, you have received the information from Sophien. Wheat is really a complicated crop, but we are not giving up. We developed a protocol which actually, first developed by, we optimized it, first developed by Pankaj Bhumik, microspore based, culture and, delivering the CRISPR system into the microspore and then do the transformation. So we are obviously at the beginning stage, not big, result yet. And we have also started some work on bacteria, fish pathogenic bacteria. So hopefully we shall proceed on. But the coronavirus is really a big threat and restricting some of our workers not to participate in the lab. Full members are now working in the lab during the coronavirus. All our universities are closed for indefinite period of time.
Yes, sir. Thank you so much for answering all of those questions. Sam, do you have more questions from these live channels?
Most of the questions were actually answered by Dr. Islam, sir. And also in the presentation. We don't have a lot of questions remaining. Someone asked that they have to use CRISPR technology in peanut plants. Is it possible mutant plants screening T1 generation or T2 generation? Something like that. I guess it was not answered. Or it was in the chat box. I don't quite make up what they tried to say. It was from YouTube chat. So we will try to answer all the questions later on in a PDF format. So we will try to compile everything that was put together in the seminar, and we will try to distribute a PDF version from our side. So I guess we don't have a lot of questions remaining. It's been like 3 hours and 16 minutes. We are doing it, and it was very captivating indeed, and we were greatly benefited from your presentations, and we love talking to you guys, and you were inspiring.
Thank you so much.
Yep. Do you have any more questions?
I think no more questions. If audience can ask anything, please unmute yourself and ask your question, please. If you have any question, audience, please. I think no more. So I'd like to thank you from core of my heart, Dr. Sophien, and Professor Tofazzal, and Dr. Kutub, Dr. Borhan, and all of you who gave your very valuable time, and who spent really great evening. And as a student of biotechnology, myself, I learned a lot. So I appreciate this program. And from every program, every day I am learning. And I'd like to learn all over my life. Thank you so much.
Thank you.
Thank you.
Thank you, Dr. Sophien Kamoun. Thank you, Dr. Islam. Thank you, Dr. Kidividin Alimilla. And thank you, Dr. Burhanuddin, sir. Thanks all of you for being here today. Something they asked is that if Science Portability can arrange on-hand training on CRISPR-Cas, we will try to do that. If we get generous funding, then we will try to put together a training workshop that you can attend. So our next seminar is in the last quarter of November.
Yes, sir. Please. Yes, Science Portability is planning to do a workshop on CRISPR-Cas9. It's really, we are trying to collaborate some Indian companies. And I have asked Dr. Mullah already that on session he will conduct. So Dr. Mullah, if you please let your opinion about that we have discussed before. And Tofazzal Islam sir here, so I would like to request him as well for conducting a session. And Dr. Sophien as well.
Thank you.
Who would go first?
Yeah, Mullah, please, you go first, because I already officially I told this thing.
Yeah, I think that would be a great chance to share the knowledge. You know, it's not share the knowledge. When you teach someone and you get prepared yourself, it's better than what you are. Okay, so that's a learning experience, and I would love to conduct a session, and I would join with other experts also.
Thank you, Tofazzal Islam sir.
Yes, obviously, we need to promote this, how to say, technology, and to train our young people. So the more we shall interact and discuss in depth, it is very important. And I am advocating, nationally also, to arrange a series of workshops, a bit longer period of workshop, hands-on training or like that. So before we are, experienced enough, I cannot invite people in my laboratory for a series of workshops. But I have a plan to organize series of, workshop to deliver hands-on training in our Global Institute of Food Security, Canada and Bangladesh joint project. We are keeping enough money for doing that job. So in future, we can do the job. And as you proposed earlier that to arrange a virtual workshop in Bangla medium for the young people, I think it is also a good idea. Because then, native language people, young people can understand better. And I will be with you. And whenever you need, please knock me.
And Kutubuddin Mullah, he is also can speak Bangla very well, so that I told him to join us, and he extended his great support that he will be with us. So Tofazzal Islam sir will be with us. And Dr. Kamoun, I would like to request you, some of your students can also conduct 1 or 2 sessions, because right now we can do any hands-on or wet lab expertise. We'll do it virtually, theoretical, giving some deeper knowledge about this CRISPR. As Tofazzal Islam sir, you heard that question that our students already, they are disappointed what they will do, how they will do. And so that we would like to arrange this session very soon, maybe in December. We will discuss together to make a small curriculum for that, and we will conduct definitely.
It would be my pleasure to do so. I'm sorry, my Bangla is not very good. But,
No, sir. It's okay. We will translate, sir. No problem.
I would love to help you out.
Google will help you to translate
Okay.
from English to
Google, yeah.
Bangla.
Very good. But I just put in the chat also I want to point out to the Grow More Foundation. So I really recommend you approach them, and I will also tell them about your activities. But the Grow More Foundation is aiming at bringing CRISPR technology to developing countries, and I'm sure they can help out also by providing kits to the students and this sort of things. And I've been supporting their activities, which are really aligned with what we want to achieve here.
Thank you, sir. Thank you so much. And we'd like to conclude right now. Saim, do we have any announcement?
Our next seminar is in the last quarter of November. That is coming in from November 20 to November 13th. So stay tuned for that seminar, and you can like and follow our official Facebook page at abby.com/sciencepodabity, and you will get the updates here. And you will do the workshop as well, as you know from today's discussion, that we will try to put together a workshop. And we wish all of you good luck, and thanks all of you for your time. Have a great day. Bye-bye.
Thank you so much. Thank you, sir.